give me a thumbs up when I'm supposed to do stuff. Hello, everyone. I am Sean Gibbons, a faculty member and microbiome researcher at the ISB, or the Institute for Systems Biology. ISB is a nonprofit biomedical research institute located in Seattle, and we're an affiliate of the Providence Health System. Before I go more into my description here of today's course, we'll start with a poll. I want to get a feel for where everybody's coming from around the world. So go ahead and fill this out while I'm giving my spiel, and then we'll come back to it at the end. So a little more about ISB. We focus on some of the most pressing issues in human health, including, of course, the microbiome, which we're all here to think about today. Also COVID-19, brain health, aging, cancer, and many chronic and infectious diseases. ISB also focuses on improving K through 12 STEM education and beyond, as you see here today. In particular, by addressing inclusion and equity for students of color, women, and other historically underrepresented groups in science. You can learn more about ISB by visiting our website at isbscience, all one word, .org, as you can see behind me here. As a nonprofit, we rely on the generous support of donors. If you like what we're doing here today or what we've done for the past 4 years now, feel free to donate and support this kind of work. There's a donate link at the bottom of our website. And as today, this week, I guess, was on Monday, Indigenous Peoples Day in the United States, I'd like to acknowledge that we are here in Seattle sitting upon the traditional unceded lands of the coastal Salish people, including the Duwamish tribe. And we honor the Duwamish tribe, past and present, for our presence here. Before we begin, I'd like to also express our thanks to our sponsors that help make these 3 days possible. In particular, Applied Microbiology International, a scientific society in the UK, who's providing free membership to those who wish, who registered for the course. And that information will come to you towards the end of the course when we share your contact info if you click the box that you wanted to be a member. So that's to be determined. It'll come later. We're also supported by Illumina, by Metabolon, and by the Environmental Health and Microbiome Research Center at the University of Washington, also known as the Embrace Center, where we have a leaf blower outside. If you can't hear that, hopefully that's Zoom filtering the noise properly. Because of the support of these generous donors, we're able to provide this course and symposium free of charge to this year of about 1,000 people from over 80 countries around the planet. I think we're excited to be able to provide this kind of bioinformatic training to such a large audience. To all you who are joining us from around the world, we're grateful for your time today. We're excited for this opportunity to train this next generation of microbiome researchers from around the planet, especially in a time where there's a lot of strife and conflict and division in the world. We like to have a little oasis where we can all get excited about microbes and focus on some science. If you're on social media, please remember to share your experiences for this symposium using the hashtag #ISBmicro23, and you can tag ISB at ISBSCI. All right, moving along. So this year, we decided, this is the 4th year that we've been running this course. This course was born in the pandemic when there weren't any options for in-person events. And we found that there was so much global engagement by providing this as a virtual resource that we just wanted to continue doing this kind of a thing going forward. So in this 4th year, each year we change the theme. And this year's theme is the microbiota as a shield against infectious disease. So the microbes in your gut are another kind of wing of the host immune system, and they actually do protect us from invasive taxa, from opportunistic pathogens, and provide this shell of protection. And so that's sort of the focus. And usually we've focused the 2 days of the course, the first one on 16S amplicon sequencing data analysis, which will specifically look into a data set of C. diff patients, pre-FMT and healthy controls. And today our instructor is Nick Quinn Bowman. He'll walk you through today's course. Tomorrow, as is per usual for our course, we're going to focus on metabolic modeling, and in particular looking at how C. diff can colonize or invade an individual's metabolic environment in their gut, and that will be led by Alex Carr. So he'll walk us through the second day as our instructor. We also have a speaker today, Katherine Ramos, who will be talking about her research relevant to today's theme. And also, I'd like to thank all of our teaching assistants for their work and support today. They'll be on the Slack helping you with all your problems. Christian Diener, Alyssa Easton, Chloe Herman, Noah Rappaport, and myself. Additional support comes from Dominic Lewis, who's our coordinator in chief. He's orchestrating everything behind the scenes. Also, Allison Kudla, Joe Meister, Audrey Hubbard, Connor Kelly, Victoria Uhl, Shauna Braga, and Thea Swanson. Thanks to all those folks for all the hard work they do behind the scenes to make all this go off without a hitch. So at this point, if we can bring up the poll results, we can kind of look at where everybody's coming from around the planet. Click on the poll. Okay, I don't see it actually. It did disappear on me. So I don't know what's going on. Maybe we won't look at the poll. Oh, wait, now it popped back up for me. Okay. So, okay, North America by far wins, which is to be expected. 67% of folks coming from North America, 12% from Europe. We have 3% representation from South America. Asia comes in at 10%, Africa at 7%, and Australia 1%. Again, nobody from Antarctica this year, but that's to be expected. So I think this shows you like who's awake right now across the planet. We tend to see that we get people in our close to our time zone tend to be the ones that join us live, and those who are asleep right now, they wake up in the morning and they'll watch our YouTube live stream later on. So that's another benefit of the way we've designed this course is it can be taken asynchronously. It's on demand. So after all of this is recorded today, you can go back and watch all the videos on YouTube and use all the course materials through the website, and that won't go away. That'll just stay up forever. All right. So if you have any issues with course materials or need assistance during the course, you can visit our course 2023 Slack channel. You've already received the link in an email. There were some people who had issues with their links. We'll have another link that we're adding to the chat, I believe, in the Zoom, so you can use that instead if that's not working, or reach out to us via email if you're having trouble. To ensure that everyone can enjoy the course in a safe, harassment-free environment, we provided a code of conduct, which is on the GitHub. And it can also, I believe, be found in the general channel of the Slack. We'll post a link to it there. If we observe behaviors that violate this code, we'll warn the perpetrators. We reserve the right to remove users from the Slack account if that behavior persists. This has never been a problem in the past, but we're just saying if people are being disruptive and preventing us from doing the education and science we want to do, we reserve the right to remove those folks. All right, and I think the last little piece is this year, we're saying no soliciting, please, in the Slack channel. If you have products to advertise or links to put out to other events or things like that, maybe please don't do that. Just kind of keep focused on the course, on your questions for the exercises. And there's a random channel for sharing emojis and fun stuff, but please no soliciting. All right. So with that, we will take a short break. The course will actually start at 9:30, so in about 15 minutes from now, Nick Quinn Bowman will come up here and begin teaching. Thank you all again for coming, and I'm looking forward to a really great day.
All right, welcome everyone to the first day of the ISB Microbiome series for this year. Like Sean said, my name is Nick Quinn Bowman, and I'm a graduate student in the Gibbons Lab, and I'll be your instructor for today's part of the course. I'm so glad that you could join us here today for the course, and I hope that you'll also join us after the course for a wonderful talk, as well as for the next 2 days of the microbiome series. We have another part of the course tomorrow, and we'll finish up on Friday with our symposium, with several talks from some wonderful folks in the microbiome field that have very exciting research to share with you all. That being said, let's go ahead and get started with today's portion of the course, which will focus on amplicon sequencing data analysis using CHIME 2. First, let's make sure that everyone is set up with the necessary materials for the course. The first thing that you'll need is the slides that I'm looking at right now. The slides are available to you in the introductory email that was sent. And the TAs will also be posting a link to the slides on the Slack channel soon if they haven't already. So I encourage you to open these slides and follow along as we go through instruction today, as well as going back through the slides if you need a refresher on any parts that you might have missed. So if you have any trouble opening the slides up, please go on Slack and let the TAs know. They'll be more than happy to help you out in finding those slides. I'll touch quickly on sort of how the course is organized. As with previous years, there's 3 main components that make up the course. First and foremost, we have the lecture slides that you should have just been able to access. This is where we'll talk about the logic behind some of the methods that we'll be working through today, go through some explanations of the processes that we'll be analyzing, as well as talk about pertinent information and link out to some of this information for you to peruse at your own time. Additionally, we'll have the interactive course notebook. In this notebook, we'll go over some of the technical aspects of the course, and you'll be able to actually follow along with us as we analyze data in real time. We'll be going through processing data, analyzing it, and creating visualizations that will let us learn a little bit more about the data that we're working with today. This is also where all the materials that you'll need to run the course will be located. Last but not least, we do have a chat for the course. You should have been able to access the Slack channel, either via the introductory email or the link that was shared on Zoom earlier. This is a great resource for you if you have any questions or need any technical support. We have several TAs on this end that are ready and waiting for you to help you with any questions. As a reminder, the Zoom chat won't be monitored as actively as the Slack chat. So if you do have any questions, I do encourage you to go to the Slack for any help. All right, so as I mentioned, all the analysis that we'll be working on today will take place in our interactive notebook. So if you have these slides open, you can go ahead and click on this link to access that notebook. A link is also available in the email that was sent to you as well as on Slack. So let's go ahead and click this to open the notebook and switch to that and get ourselves set up. I'll give you all a second to open up that notebook. And hopefully you all see something that looks like this. So this is our interactive notebook that we'll be using, again, to run through the actual data analysis component of today's course. Before we begin, though, I would encourage you to make a copy of this notebook in your own Google Drive. That'll allow you to save your work and come back to it later on if you so desire. So to do that, we'll go to this toolbar on the top left side of the page, click on File, and about halfway down, you'll see an option that says Save a Copy in Drive. So go ahead and click that. If you haven't logged into Google, you may be prompted to do so. And this will save a copy of the notebook directly into your Google Drive. And you'll see on the top left, it says Copy of the notebook. So we can rename that to be something a bit more meaningful. For instance, I'll just call it Nick. Great, so now we have our own copy of the notebook. But what exactly is the notebook? What are we looking at here? Well, you can kind of imagine this to be an interactive document that, again, will allow you to run through the data analysis components of the course. The notebook is divided into different cells, some of which contain text, like this first one that's highlighted here, and some of which contain actual code that we'll be running. So those are the cells that are a little bit darker gray, and they'll have this play button at the very on the left-hand side of the cell. Let's familiarize ourselves a little bit more. So on the left-hand side, we see these 4 icons on this panel here. The first icon will open a table of contents, and that'll kind of let us know what we'll be working through today and the general workflow of our course. A little bit more exciting, though, is this last icon down here. So this is our file directory. And right now we see that there's nothing in it except for some sample data that we won't be using. So let's go ahead and fix that. So the first cell that we'll run in our notebook will actually copy in all the materials that we'll need for the course today directly from GitHub. You'll notice that many of the cells in our notebook start with this exclamation point. And so the reason for that is that our Colab notebook that we're using is actually running Python code as a default. A lot of the commands we'll be running today are actually meant to be run in the command line. So to tell the notebook that it should expect a command line action, we'll prefix the bit of code with an exclamation point. So let's go ahead and run this. And so this will save this course materials directly onto the Google Colab server and won't copy anything to your local machine. So don't worry about that. And this should only take a couple minutes to copy, or a couple seconds to copy all of the materials over. Right, so now if we look at our file directory on the left-hand side, we should see a new directory called materials. If you don't see that right away, you can press the second icon here, and that'll refresh your file directory. If we open up the materials directory, we can see a whole bunch of stuff that was just downloaded that we'll need for the course, both today and tomorrow. Now, in order to actually use this material, let's change our active working directory to be this materials directory. And that's what we'll do in this next cell. So if we run this cell, the working directory will change to be materials, and that just will make it easier for us later on to find our relevant data and the resources that we'll need. Great, so then now the last thing that we need to do in order to set up our notebook for us to use it for analysis today is to install CHIME 2. So as you might remember from the title of today's course, we'll be using a computational library today called CHIME 2. And that involves, using CHIME 2 involves downloading it and then installing and setting it up within our notebook. This process itself will take about 10 to 15 minutes. So let's go ahead and run this cell. So it'll start in the background. And again, this will only set it up in the Google Colab notebook, not on the local machine. Okay, so like I said, this will take a little bit of time. So as the notebook finishes setting up for us to use later, let's switch back over to the presentation and discuss a little bit of background information about what we'll be doing today and some of the implementation. And again, if you run into any issues with setting up the notebook or accessing the notebook, please don't hesitate to ask on Slack. The TAs will be more than happy to help you. Great. So now we're back onto the lecture slides. One more thing I'd like to mention, if at any point during the course today, you lose access to your notebook or you somehow aren't able to complete one of the cells and you are missing any output that's necessary to complete the rest of the notebook, all of the outputs we generate today can be found in the treasure chest folder. This is available both at the GitHub that's linked out here, as well as in the materials folder that we just downloaded into our Google Colab notebook. So if you are missing anything, that can be found in that treasure chest. Again, if you have any issues with that, just ask the TAs. They'll be happy to help you. Great, so now we'll start a little bit of background. By virtue of you being here today, I assume that you're somewhat familiar with what the gut microbiome is, but just in case, we'll have a quick little refresher. So when we talk about the gut microbiome, we're talking about the 30 to 40 trillion bacterial cells on average that live in the human large intestine. Specifically, these cells live in the mucus layer that lines the large intestine. And importantly, they're very heterogeneous between individuals. That is, you and me and everyone else in this course has a very unique and distinct set of microbes that live in our large intestine. So what exactly do these microbes do for us? Well, there's a number of things. One of the primary things that they do in our body is they help us digest food. So things like resistant starch or cellulose that our body can't break down makes its way into the large intestine and is consumed by these bacteria. They'll then produce other metabolites that can go back into the body and have a number of either positive or adverse health effects, from mitigating or modulating inflammation to increasing our risk for cardiovascular disease. And that's not all that the microbiome does in terms of affecting our health. It's also involved in training our immune system, modulating our mood through the gut-brain axis, which is still being fully understood, and determining how susceptible we are to pathogenic invasion of our microbiome, which we'll talk a little bit more about throughout the duration of the course. So as we can see, the microbiome itself does significantly affect our health, and the composition of bacteria that are present in this community is very important in terms of dictating sort of our overall health trajectory. So this might lead you to wonder, how do we know what's in someone's microbiome? How can we actually measure and determine what microbes are present in any one person's microbiome? So to do that, that's actually not a trivial question. First and foremost, there's hundreds to thousands of taxa in each individual, and it's actually fairly difficult to culture these taxa outside of their resident environment in the large intestine. So instead, we can do what's called sequencing of their DNA instead. So we take a fecal sample from any individual and extract the DNA, read that DNA, and use that to determine what microbes are present and in what abundances. But this in itself raises a few questions as well. One, how do we go about getting the sequencing data? And secondly, once we have that sequencing data, how do we process it and analyze it? Oops. So to answer that second question first, I'd like to introduce you to a computational tool called CHIME. So spelled Q-I-I-M-E, but pronounced chime like a wind chime. This is a tool that's meant for the processing and analyzing of microbial sequencing data in order to make that process a little easier and more transparent. So CHIME was created around 2010 during the Human Microbiome Project under the leadership of Drs. Greg Caporaso and Rob Knight. And with the release of CHIME, what they sought to do was take this process of going from raw sequencing reads and transforming them into a format that allowed us to get meaningful outputs from that raw sequencing data and streamline that process, make it a little bit more easier for researchers to conduct and more transparent in terms of determining exactly what transformations were made to this data. And that's exactly what they do with CHIME. Around 2016, CHIME 2 was released, which is a new implementation of CHIME that introduced new semantic types to CHIME, as well as more of an emphasis on analysis transparency. So what exactly is CHIME that we're talking about here? Well, to borrow a phrase directly from the CHIME 2 documentation, CHIME 2 is a powerful, extensible, and decentralized microbiome analysis package that has a focus on data processing and analysis transparency. So sort of like we already talked about. And I would say that's a very good description of exactly what we can do with CHIME. But in terms of actually using it, what can we do with CHIME and how do we implement it? Well, essentially, CHIME is actually just a set of commands that we can use to transform raw microbiome sequencing data into intermediate outputs, as well as visualizations that give us context and information about the communities that we're looking at. As you can see in the little GIF here, it's most commonly used via the command line. Today, we'll actually use it within the CoLab notebook, but it's actually very straightforward to use and allows us to streamline this process of going from raw sequencing data and transform that into meaningful outputs that we can use to learn a lot more about our microbial communities and visualize them as well. So before we talk a little bit more about CHIME, I'd like to mention that CHIME 2 itself comes with a lot of help. So if you are interested in using this outside of today's course, there's a large range of tutorials that'll teach you more about individual parts of the CHIME 2 pipeline, general documentation that will help you understand more of the methodology that's used, as well as a very useful user forum where you can go to ask questions or take a look at questions that have already been asked. Today, we'll go through sort of a basic version of the CHIME 2 pipeline, but there's a lot of really cool and powerful things that you can do with CHIME that we won't touch on all of that today. And so I do encourage you to take a look at those tutorials and the documentation if you are interested in using this more for your own purposes. Great. So before we jump into actually using CHIME, I think it's important to touch on how CHIME 2 is organized. So generally, CHIME 2 manages what are called artifacts, which are essentially intermediate data files that we can use to store the output of different CHIME actions, which then feed into other methods within CHIME. It also produces what are called visualizations, which are essentially endpoints of the CHIME 2 pipeline that allow us to infer or draw conclusions about our data. For instance, on the right-hand side, we can see an example of the CHIME 2 pipeline that we'll be following today. So first, we start with our raw sequences. In this case, green rectangles represent CHIME 2 artifacts. So our raw sequences we import as an artifact. That then feeds into a method, which is a yellow rectangle. For instance, demultiplexing your data. Today, we won't actually demultiplex our data, but if, for instance, you had multiple samples within a single sequencing run, demultiplexing would separate those samples out, and so we could isolate them individually. This demultiplex function then creates a new CHIME 2 artifact, demultiplex sequences, which goes into another CHIME 2 method, and so on and so forth. At the very end of the pipeline, we'll see that there are also these blue boxes, which represent visualizations. So again, visualizations are, rather than being, excuse me, intermediate files, these are actually endpoint files that allow us to take a look at statistics or perhaps graphs that describe our data and allow us to draw conclusions and gain some ecological insight about the communities that are being sequenced and looked at in these cases. So one thing to remember is that, like we touched on, artifacts are really just meant for, artifacts represent intermediate steps. So we don't necessarily use artifacts to look at directly in terms of drawing conclusions about our data. Instead, for that, we use visualizations, which are endpoints meant for human consumption. For this reason, though, visualizations cannot be used as inputs for additional commands. So we can't go ahead and build a visualization and then plug that back into the CHIME 2 pipeline. So this was generally just a high-level description of what CHIME 2 is, how we might go about using it, and its general structure. And as we go through the course, we'll get a little bit more technical about using individual CHIME 2 commands and how we go about doing that. But if you recall the question that I posed earlier, this only answers one of those two questions, which was how do we process and analyze microbial sequencing data? The first question I asked was, how do we actually go about getting that sequencing data? And so let's take a look at that, which I think is very important considering that this is sort of the basis of all the data analysis that we'll be using, that we'll be running today. So when we talk about sequencing microbial communities, there's really two primary methods that you might use. First, and the method that we'll be using, or that generated the data that we'll be using today, is called amplicon sequencing. So in this case, after extracting DNA from our samples, rather than sequencing all the data, or excuse me, all the DNA that's present in the sample, we'll actually just focus on one small part of a gene that's called a marker gene. This marker gene is somewhat special because it has two very important parts. One is a small conserved region that's identical between all of the taxa present in our samples. We need this conserved region in order to add a PCR primer that will attach to that conserved region and extend a DNA read. After the conserved region, we also need what's called a variable region. So these are unique to the individual taxa that are present in our samples. So after extracting DNA, we'll add our PCR primers that will attach to those conserved regions and amplify that one small part of the marker gene. We can then sequence just those PCR amplification pieces and use the sequencing results of that variable region to go back and determine where each piece of DNA came from. As you can see, by just sequencing a much smaller part of the genome, we can minimize the amount of sequencing and computational labor that's required. In bacterial communities, we most often use the 16S gene as our marker gene. And we use specific regions of that 16S gene as our conserved and variable amplification pieces. Another option that you might use for sequencing microbial communities is what's called shotgun metagenomics. So in this case, after extracting our DNA, we'll cut it up into smaller bits in a process that's called fragmentation. And then essentially using random PCR primers, we will sequence the entire genome. So all the DNA that's present in our samples. And at the end, what we'll end up with is small pieces that cover the full genome of each taxon present in our sample. This can be pretty useful for certain kinds of analyses. For instance, if you're looking at functional metagenomics, so the genomic capacity of the entire community. But as you can imagine, this also is much more intensive in terms of the amount of sequencing that's required, as well as the computational effort that's required. It ends up being much more expensive. So for the data that we're looking at today, we'll be focusing only on the left-hand side, which is amplicon sequencing. Like I mentioned, for bacterial communities, we most often use the 16S gene as our marker gene. So why is that? Well, the 16S gene, importantly, codes for a subunit of the ribosome. And since we know all bacteria need ribosomes in order to survive, we can safely assume that this gene is present in all of the taxa within our sample. Additionally, the 16S gene has that morphology that we were talking about previously. It has conserved regions that are shown in blue that are nearly identical between all of the taxa in our sample, and which we can use to attach a PCR primer. It also has interspersed between those conserved regions, hypervariable regions that are shown in pink that display phylogenetic heterogeneity. That is, they are very specific to the taxon that they come from. Most often, we use either the V3 or V4 hypervariable region. The data that we're using today actually does sequence that V4 region of the 16S gene. Right, so that was just a general overview. But now let's consider and talk about what we'll actually be working on today within our notebook. So the theme of this year's course was how the ecology of our gut microbiome protects us from invasion by pathogenic bacteria. And today we'll actually be analyzing the microbial composition of the gut microbiome of individuals with recurrent C. difficile infections, or C. diff for short. C. diff is an opportunistic pathogen that will come in often after antibiotics are taken by an individual, invade the microbiome, and can lead to all sorts of pretty nasty symptoms, from diarrhea and inflammation to, in severe cases, internal hemorrhaging and even death. It's also really difficult to get rid of. Adding more antibiotics can lead to what's called a recurrent infection that starts a cycle of reinfection. Interestingly, it seems that the composition of one's gut microbiome can dictate how at risk you might be for the invasion of C. difficile. And in fact, some people can actually carry C. difficile asymptomatically for years, so they don't have any of these bad symptoms. And we'll talk about this some more in tomorrow's part of the course with Alex as well. So today we'll be looking at 16S amplicon sequencing from that V4 region of the 16S gene from human fecal samples in 4 healthy donors that do not have a C. difficile infection, as well as 4 individuals that do have recurrent infection of C. difficile. And we'll be comparing how the composition of those microbiomes sort of varies between folks. if you're interested in learning more about the data that we're using today, you're welcome to follow the link that's shown on the screen here, and that'll take you to a publication from which these data are collected. So when we talk about C. difficile, like I mentioned, typically C. difficile presents itself in individuals that have taken antibiotics for one reason or another. And it's more common in folks that have also been staying in a hospital. If you combine that with a weakened immune system, and as well as possibly a previous infection, you drastically increase your risk of contracting and getting infection of C. difficile. And so we can quickly take a look at the infection cycle and look at how recurrent C. difficile infection typically presents. So like I said, you might be given antibiotics for one reason or another. You have a bacterial infection. And those antibiotics are meant to wipe out the bad bacteria, which they might, but as an unintended consequence, they might also wipe out a bunch of your good gut microbes that are living in your gut microbiome. What this does is essentially opens up a bunch of real estate for C. difficile to come in and take over the microbiome and make you pretty sick, which is what we see in this individual with the sad face. An easy approach that you might consider to get rid of C. difficile is to add more antibiotics, which would kill off the C. difficile. But you can also see how this would essentially just open up more space in the gut microbiome and open someone up to recurrent infection. So it's a pretty tricky problem to solve. One therapy that actually has proven very successful in treating C. difficile is what's called a fecal microbiota transplant. So in this case, we take a fecal sample from a healthy individual, isolate the microbiome, and engraft it in the microbiome, engraft it into an individual with a recurrent C. difficile infection. Over time, this engrafted microbiota will expand and essentially push out the C. difficile. And hopefully lead to, again, a healthy and balanced microbiome. And FMTs are actually largely successful in treating FMT and are one of the only therapies that really is effective in doing so. Okay, so now we have an idea of what sort of data we're looking at. Let's take a look at the overall workflow of what we're actually doing today as far as our analysis. So we will start with raw sequencing data that comes from the amplicon sequencing we just discussed. And we'll feed that into our CHIME pipeline. So CHIME will go ahead and trim and filter out low quality sequencing reads. It'll also then go through a process in which it infers sequence variants, essentially just finding the most likely set of original sequences that were present in our original sample. We'll then also assign taxonomy to each of those sequencing reads. So we know the identity of the taxa present in any individual's microbiome. And then after some data normalization, we'll go ahead and look at some metrics that describe our communities in terms of diversity and gaining more insight into the ecology of the microbiome. First and foremost, we'll have to import our raw data into CHIME as an artifact. So if you recall, CHIME uses artifacts. And so that'll be our first step. So let's get started, and we'll switch back to the notebook and get started with that. Okay, so at this point, we're back in the notebook. Hopefully, your setup of CHIME 2 has concluded and it all worked well. If you have any issues, please ask the TAs. They'll be more than happy to help you. And now we can actually get started with using our notebook for data analysis. So like I mentioned, we want to start with our raw sequencing data. So let's familiarize ourselves with that. If we go to our left-hand side in our file directory and open this data folder, we'll see that we actually have what are called FASTQ files for each of our 8 samples. This is the file type that typically comes out of sort of raw sequencing runs. And we see we have all 8 of our samples. We also have 2 files at the bottom, one called a manifest and one called metadata. And these are both tab separated value files that we can very easily read using a library called pandas. So if we import pandas, let's go ahead and take a look at what our manifest file looks like. And this will only take a second. So here we see our table containing our manifest. And essentially, this is just a map that we'll give to CHIME that will allow it to find and import our raw sequencing files. It's very simple. We just have one column that identifies the sample and a second column that identifies where that sample is located. So we'll use this in our first step in CHIME, in which we'll import our raw sequencing data as a CHIME artifact. Let's also check out the metadata file just to get an idea of what that looks like. So essentially, the metadata file, which is also in this data directory, just gives us more context about our samples. So we know we have 4 healthy samples and 4 samples with recurrent C. difficile infection. And we can see that in this disease state column over here. There might also be other associated metadata in terms of age, height, and maybe body mass index. And we'll use this later on when we want to actually compare samples between these 2 groups. So that looks good. Looks like we have all 8 of our FASTQ files. And now we can use these data to import our files directly into CHIME. So let's go ahead and start our CHIME 2 pipeline. The first thing we want to do, like we've mentioned, is import our data. That's what this cell here will do. So every time we use CHIME, the command will take a very similar format. We start with our exclamation point, since this is a command line action. Then we'll call CHIME as our library that we're using. CHIME has several different plugins, which are essentially libraries that allow us to do different things within CHIME. In this case, the plugin that we're using is called Tools, which is sort of general, but allows us to import our data. Finally, we'll specify the action we want to do, in this case, import. Like I mentioned very briefly, so you might have missed it, the release of CHIME 2 included the introduction of semantic types, which essentially means it specified what kind of data needs to be put into each portion or each part of the CHIME 2 pipeline. So in this case, we'll tell CHIME what type of data to expect. In this case, since we're importing our raw sequences, we're looking at sample data as the type. We'll also specify our input path. So again, we'll be using that manifest file as sort of a map to tell CHIME these are our samples, and this is where the sequences can be found. And then we'll also specify an output path. In this case, the output path is a CHIME artifact that we'll call sequences. All CHIME artifacts take on a file name that ends with .qzv. That stands, or QZA, excuse me. That stands for CHIME zipped artifact. We'll also tell CHIME the format of our sequencing data, which in this case is single end, since we only sequenced in one direction, as opposed to paired end, where you sequence in 2 directions. As well as their FASTQ files, and we're importing using a manifest. So let's go ahead and run this cell. And this should only take about 20 to 30 seconds. And in the end, what we'll see is that we should have a CHIME artifact called sequences.qza, which we can then use to start our CHIME pipeline. And this will show up on the left-hand side in our file directory in just a second when this cell finishes, and it looks like it's just about done. Again, if you don't see it right away, you can hit this refresh icon, and you should see it near the bottom here, sequences.qza. Great, so now we have our sequences, and we can theoretically move forward with the pipeline, but often you want to double check that at intermittent points in the pipeline, we want to double check that our data kind of looks the way we would expect it to. And in this case, we might be interested in looking at whether or not our sequencing data looks like it's of good quality. Right now, though, all we have is a CHIME artifact. And if you recall, artifacts aren't really meant for us to take a look at directly. So instead, what we want to do is use that artifact to build a visualization. And that's what we'll do in this next section here. So if we run this cell, essentially what we're doing is we're calling CHIME again, and we're using the demux plugin. So demux is meant for demultiplexing sequences, which, like we mentioned in the slides, we won't actually do. But within the demux plugin, there's a tool called summarize that we will be using. So here we'll tell CHIME to take our input data, which is this sequences.qza file, and build a visualization, which we'll call qualities.qzv. So let's go ahead and run this cell. And yeah, this shouldn't take long. Importantly, visualizations in CHIME all take on a file name that ends with QZV, so CHIME zipped visualization. So that's how you can identify between artifacts and visualization. This is the end of the file name. So how do we actually look at these visualizations though? Well, unfortunately, we can't look at them directly within our Google Colab notebook. But luckily, CHIME actually does offer us a very useful tool that we can use to look at our visualizations. So when this finishes here in just a second, looks like it's about done. We'll go to our file directory, locate the visualization we just constructed, qualities.qzv. And if we go to the right-hand side of this column, you'll see 3 little dots. Click on those and then download the file. And this will download it locally. Now, if we want to take a look at that visualization, we can open view.chime2.org. And you'll find a link to that within the notebook as well. So if we go to view.chime2.org, we'll see something that looks like this. And there's this big gray rectangle that says drag and drop or click here. So what we want to do is import our visualization that we just downloaded. So I'll go ahead and click on qualities.qzv that we just downloaded, open that, and right away we can see our visualization. So congratulations, we've now made our first CHIME visualization. Visualizations don't always take on the format of a graph or a heat map or anything. Sometimes they also look like this, and they have data tables in multiple parts. So let's take a look at what this is essentially telling us and determine if our sequencing data is of good quality. So first and foremost, we see this summary of our sequencing counts from each of our samples. At minimum, it looks like the sample with the fewest counts is about 25,000 reads, and our maximum is well over 100,000 reads. As a general rule of thumb, we want at least 10,000 reads per sample to be confident in our sequencing. So this should be more than enough for our purposes here today. We'll also see this data represented as a histogram here with all 8 of our samples, and then also a table that just lists the number of reads for each one of our samples. In this case, since we did single-end sequencing, we don't have any reverse reads to show. If you did paired-end sequencing, you would see a similar histogram and table for those reverse reads. So this is great, but it doesn't really tell us about the quality of our sequencing, just the number of sequences we have for each sample. To really get at the quality, we'll go to the top left side here and click on the second tab that says Interactive Quality Plot. And this is the more interesting part, in my opinion. So here we can see what is essentially a graph describing the quality of our sequencing as a function of the length of a sequence. So on the x-axis here, we can see the number of bases in each sequence as it gets longer, with a maximum length of 151 bases. So the longest read in our data is 151 bases long. On the y-axis, we have a quality score, which is a little bit ambiguous at first, but essentially tells us the error rate, or the estimated error rate of our sequencing results. So every time we sequence a piece of DNA, there's a chance that the sequencer will make a mistake and assign a base incorrectly. So it might assign an A when it should have assigned a T, or for instance. And that's what this quality score describes. In general, a higher quality score is better. And a good rule of thumb is that a quality score of 10 correlates with an estimated error percentage of 10%. So in that case, 10% of the reads in your sequence would be labeled incorrectly. It decreases logarithmically though. So a quality score of 20 corresponds to an error rate of 1%, 30 of 0.1%, and a quality score of 40 indicates an error rate of 0.01%. So generally, any quality score between 30 and 40, we can feel very good about. Between 20 and 30 is still pretty good. We can feel relatively confident about that. As it starts to get lower towards a quality score of around 10, that's where we don't really want to be using those data anymore, because we can't feel confident about the quality of those base assignments anymore. So how do we use this data and the results from this moving forward? Well, within CHIME, one of the steps that we'll be following is called trimming our reads. So using this quality score graph, we can determine where along this x-axis we'd like to trim our reads, meaning that we'll only use reads that are at that level or shorter, and we'll essentially get rid of any reads above a certain level. So based on this graph, maybe some of you can post on Slack, and if you have any ideas of where on this graph you might truncate your reads. So what sequence space you might choose to cut off everything to the right of that. And if anyone has an idea, yeah, maybe put it on Slack. And while you do that, I'll say a couple more things about this plot. You often see that the sequencing does, the quality score does decrease as a function of length. So typically as the reads get longer, sequencing, quality score, excuse me, gets a little bit worse. You often also see this sort of relationship where the first couple bases seem to be of poor quality as well. And that's actually just an artifact of how quality score is calculated, since it takes into account preceding bases as well. And obviously there's no base before base number 1, so the quality score is artificially a little bit lower there. You can also see down here a 7-number summary for each of the positions within our graph. So if we zoom in on a piece of the graph, we can do that by clicking and dragging, we'll see these are actually box plots that describe the quality score at each position. And if we click on one, we can see the 7-number summary showing the sort of upper and lower trends of quality score at that position. We double click on the graph, we zoom back out. So do we have any guesses on Slack for what a good truncation length might be? Great, yeah, so these are all good guesses. And yeah, so keeping in mind, we want to cut off essentially any bases that are of low quality. So in this case, like I said, anything above 30, we can feel pretty confident about. So since only our last base really falls significantly below a quality score of 30, we'll go ahead and truncate at a length of 150. So we'll only get rid of that last base. And quite frankly, with data that looks like this, you really don't have to worry too much about truncation, since most of the bases are at pretty good quality. But it is important to remember, if you're working with your own data, and maybe the read lengths are a little bit longer, to consider that as the read lengths get longer, the quality scores seem to dip a little bit. So always remember to take a look at that and consider that when you're deciding where to truncate your reads. Okay, let's switch back over to the notebook and see how we can implement that truncation length. Great, so now we have our data as, our sequences as a CHIME2 artifact, and we can use it to start that CHIME2 pipeline. The first step in the pipeline, as you might remember, is this filter and trim step. So we're essentially filtering out low quality reads. And this step is called denoising. And to denoise our data, we'll use a plugin that's called data2. And this does a number of things for us. First, it filters and trims the reads. So like we just discussed, it'll trim the ends off the reads that are of low quality. It'll also remove any reads that are just of low quality overall. It'll also go through this process of identifying ASVs, which we'll talk about more in detail when we switch back to the slides. It'll remove chimeras, which are artifacts from PCR implication. And then finally, it'll go ahead and count the abundances of each of the ASVs that it identifies in step 2. So obviously, this step is doing a lot. So this ends up being one of the longer steps that we'll have to do. So let's briefly take a look at what this cell is doing. Then we'll run it and switch back to the notebook while it continues, because it usually takes about 10 to 12 minutes. So while that's running, we'll switch back over to the notebook. So first, let's look at the cell though. We're calling CHIME, and within CHIME, the data2 plugin. And the specific action is called denoise single. Single, again, because we're using single end sequencing as opposed to paired end sequencing. We'll give it our sequences. So this is the CHIME artifact with our raw sequencing data. And we'll also identify that truncation length that we just chose from our quality scores. If you chose different truncation length, this is where you would input that. But I recommend for now to stick with a truncation length of 150. We'll also identify the number of CPUs that we want the the server to use. In this case, 2 is the maximum we have available to us, and this will increase the speed of computation. Finally, we'll tell it where to put all of our outputs from this process. So the denoise process actually produces several artifacts rather than just one. So we'll specify a directory that we want it to put all of the outputs into, and we'll call that data. We'll also add this verbose flag, so it'll essentially just tell us what it's doing at each step of the process. So let's go ahead and hit play. And so this will start running. And again, this will take about 10 to 12 minutes to get all the way through. And so while that's going, let's switch back over to the slides and discuss in more detail what actually we're doing in this step, as well as some of the additional implementation that'll come after. All right, so hopefully now you can all see the slides. And yeah, so what we just started was a process called denoising, using data to our CHIME 2 plugin. And like we just mentioned, this is doing a number of things: filtering and trimming, finding ASVs, removing chimeras, and then finally counting the abundances of those ASVs. So this is our review that we just discussed. But let's take a look at each of these steps individually. All right, so that first step, filtering and trimming. This is doing essentially 4 separate things. First, we're trimming low quality regions. We just discussed this. We identify our truncation length, and we'll trim all the reads beyond that truncation length, which are of low quality. Second, it'll also remove any reads that are of low average quality throughout. So if some of our reads just have a bad quality score from the start, those reads will be removed. It'll then also remove reads that have what are called ambiguous bases. So sometimes sequencers have trouble identifying a base in the sequence. And so it might say, well, I know there's a base there, I don't know what it is. So it'll add to the sequence an N, which is considered ambiguous. So CHIME will remove that since it's not really helpful to us moving forward. Lastly, it'll also remove PhyX, which is a bacteriophage genome that's often added to sequencing to make the quality a little bit better. It essentially just increases the diversity of a sample, since a really homogeneous sample can can confuse the sequencer and lead to poor sequencing quality. But we don't need it moving forward for our analysis, so CHIME will remove PhyX from our sequence reads. Step 2 is identifying amplicon sequencing variants, or ASVs. So this sounds kind of complicated, but it really boils down to exactly what we said earlier, which is we want to identify which sequences represent the sequences that were present in our original sample. So like we just discussed, every time that we sequence a piece of DNA, there's a chance that the sequencer will make a mistake here or there. So at the end, we might end up with several thousand unique sequences that might look similar, but vary in different positions here or there, due to these errors that the sequencer can make. As it turns out, the errors that the sequencer makes, so identifying or mislabeling a base here or there, the probability of those errors occurring is actually a function of the quality score, where on the sequence the error occurs, as well as what kind of error is being made specifically. So the probability of an A becoming a T might be higher than the probability of an A becoming a G, just for instance. So, and this can be described by what's called an error profile, which we kind of see on the top right side here. If we knew this error profile ahead of time, it would actually be fairly easy for us to look at those sequence variants and identify where the most likely errors were that occurred, and work backwards from this to find a corresponding set of the most likely set of sequences that represent the sequences actually present in our original sample with no errors. Alternatively, if we knew what those sequences were, those original sort of ground truth sequences, it would be very easy for us to calculate the error profile. Unfortunately, in practice, we don't know either of these things ahead of time. So Data 2 uses what's called an expectation maximization algorithm to calculate the real error model and find the corresponding set of actual amplicon sequencing variants at the same time. So essentially it does this by first taking a guess at what might be the sort of ground truth amplicon sequence variants. So the real sequences with no error. So for instance, if it finds a set of reads that show up several thousand times in the sample and look identical, it might assume, okay, this might be one of the sequences that was actually present in our original sample. It can then use that to learn and calculate an error profile. And then it continues to iterate this process back and forth until it converges on the true error model of the sequencing data, as well as a corresponding set of amplicon sequencing variants that actually represents the most likely set of sequences that were present with no errors in our original model, or in our original sample, excuse me. And this is actually a pretty cool and robust algorithm that works very well. And at the end of the day, you might start with several thousand different sequence variants with a few changes here and there. And Data2 will be able to sort of converge those down into a set of 50 to 100 ASVs that are specific to the original sequences that were present in our sample. This is a pretty cool trick that we can use to identify those ASVs and end up with a set of representative sequences that represent the taxa present in our data. The third thing that Data2 does is it looks for what are called PCR chimeras. It looks for them and removes them. So PCR chimeras are an artifact that comes out of sequencing, wherein the primer attaches to that conserved region of the 16S gene, and it starts extending through the hypervariable region. But for one reason or another, that extension is aborted. So now we have this unfinished piece of DNA kind of floating around, and it might then find a similar piece of DNA, but importantly a different one, and misprime or attach to that piece of DNA, but then continue that extension. And so what we're left with at the end is a chimeric piece of DNA that's half one, that's coming from two separate templates, that doesn't represent any of the DNA present in our actual sample. And so we ideally want to remove these, since these can lead to additional artifacts later on when we're trying to design taxonomy and can confuse our data in general. So Data2 will go through and identify and remove these chimeras from our sequences. The last thing it'll do is then count the abundances of each of the ASVs in our sample and return to us a list of these ASVs. So these are now sort of the abundances of each of these representative sequences. To this point, though, we don't actually know the identity of any of these ASVs. We just know their representative sequence and how abundant they are in each sample. So does anyone have any ideas for what we might be able to do with those data sets? Sort of what metrics can we calculate using only that data that can tell us something about our communities? So it may take a second to think about that and answer in Slack if you have any ideas. I'll give you a few more seconds here. All right, so unless we have any ideas popping up in Slack, totally, yeah. So I think someone mentioned diversity, and that's exactly what we're going to look at first. So when we're looking at microbial communities like these, one of the things that we're interested in using to describe these communities is sample diversity. And diversity generally comes in two separate flavors. So there's alpha diversity as well as beta diversity. So now, while Data2 is still running, we'll take a second to go through these and sort of discuss the difference between these kinds of diversity and how we use them to measure and describe our communities. Alpha diversity is pretty straightforward. Essentially, alpha diversity asks the question, how diverse is a single sample? And this can really be boiled down to two different metrics. The first metric is richness. So to determine richness, we can essentially just count the number of taxa that we observe in a community. For instance, this last community on the right side, not very diverse. There's only one taxon that we observe in that community. The second community, though, we see 3 different taxa, so a little bit more diverse. And in this first one over here, we also see 3 different taxa. So also somewhat diverse. However, we can also see that this second taxon seems to, or this second community seems to be dominated by this circular taxon. So richness in itself doesn't necessarily describe the overall diversity of our samples. Instead, for that, we'll also look at a metric called evenness, which essentially asks, how evenly are the abundances in our sample distributed across the taxa? So do we have one taxon that's, dominating the overall composition of this community? And so if we take this into account, we can see that there's more diversity happening in this first community on the left than in the central, the center community, since there's higher evenness in that community. Commonly, when we're actually measuring alpha diversity, we'll find a metric that combines both richness and evenness to paint a pretty good picture of the actual alpha diversity of our samples. So for this, we might use something like Shannon index or Simpson's index. Shannon index essentially tells us how surprised we might be at pulling out finding any specific taxon within our community. For instance, if we look at the community on the right-hand side, and we at random pick a taxon out and find a circle, and then we pull out a second taxon and it's again a circle, by the third taxon that we pull out randomly, if it's again a circle, we won't be very surprised. And so that's a relatively low Shannon score. On the other hand, if we look at this first community on the left-hand side and randomly pick a taxon, and then again randomly pick a taxon, we might end up with a different taxon each time, since there's much more diversity here. And that's essentially what Shannon index tells us. It's a metric of alpha diversity that really represents both richness and evenness. So that's one that we commonly use. The neat thing about alpha diversity is that at the end of, after you calculate alpha diversity, you're left with a single discrete value for each sample. So that is, it can be treated as any other measurement that you might take of a sample, and makes it very easy to compare samples between each other and perform many of the classic statistical tests that we're familiar with, things like t-tests or Mann-Whitney U-tests, to determine whether there's statistically significant differences in alpha diversity between our samples. Beta diversity, on the other hand, is slightly more complicated. So essentially, beta diversity asks how different are two or more samples from one another. So rather than within a sample, this is diversity across samples. And this again comes in sort of two different types, similar to the richness and evenness that we saw in alpha diversity. Unweighted beta diversity just asks how many taxa are shared between samples. For instance, this set of communities on the right, we see they couldn't be more different. There is no shared taxa between sample 2 and sample 1. On the other hand, in terms of unweighted beta diversity, the two communities on the left-hand side are perfectly similar. So they have the exact same taxa shared between each other. Obviously, we can see that the abundance of these taxa is actually different, but if we only looked at unweighted beta diversity, that wouldn't be taken into consideration. Weighted beta diversity, on the other hand, does account for the abundance of shared taxa between the samples. So in this case, we would see that there's actually a difference in weighted beta diversity between sample 1 and sample 2 on the left-hand side. Obviously, we still see a very high difference in beta diversity for the two communities on the right-hand side. So common metrics that we might measure to determine beta diversity are what's called the Jacquard index for unweighted beta diversity, the Bray-Curtis distance for weighted beta diversity, as well as unweighted or weighted unifrac distances. Unifrac is additionally special because it takes into account one more consideration, which is how different are samples in terms of phylogenetic relatedness of taxa within those samples. So we might be wondering, if samples aren't necessarily shared between, if taxa aren't necessarily shared between samples, how closely related are the taxa that are present in those samples? And so unifrac answers that. Well, how does that work? So in order to determine beta diversity while accounting for genetically similar taxa, the first thing we have to do is build what's called a phylogenetic tree. So essentially, this is a tree that clusters taxa across all of our samples based on how genetically similar they are. So how much their genetic sequences diverge from each other. So that's what we can see in these two phylogenetic trees here. On the left-hand side, we have two samples that are very similar to each other. So the taxa in sample 1 cluster very closely with the taxa in sample 2. That is, they're pretty close together on the tree. Unifrac essentially calculates this by measuring how far up the tree we have to go before we reach a node that's shared by both samples. So in the tree on the left-hand side, we can see we only have to go up one level to reach a node that's shared by by both samples. In the tree on the right-hand side, we see we have to go all the way up to the very top of the tree before we reach a node that's shared by both sample 1 and sample 2. Unifrac essentially is just a fraction of those shared branches is a fraction of those shared branches versus all the branches present in the tree. That's unweighted unifrac. Weighted unifrac, on the other hand, does the same thing, but it scales those branch lengths by abundance. So more abundant taxa are more represented in that. Great, but how do we actually go about building a phylogenetic tree if we want to look at beta diversity in terms of phylogenetic relatedness? Well, one of the things that we want to do there is we want to see how closely related all the sequences across all of our samples are to each other. And to do that, we use what's called a multiple sequence alignment, and then arrange those sequences together by similarity. And this is represented by branch length. So shorter branches indicate closer sequence similarity. Longer branches, less similar sequences. Unlucky for us, there's actually a plugin in CHIME that allows us to build these phylogenetic trees. And shortly, when we switch back over to the notebook, we'll go ahead and do exactly that. Let's quickly revisit beta diversity, though, and consider this. Once we've calculated and measured beta diversity between all of the samples, how do we visualize that and actually analyze that data? If you recall, for alpha diversity, we get back a discrete single value for each sample, which makes it very easy to compare one sample against another. For beta diversity, though, the value that we get back is actually the sort of dissimilarity between each pair of samples. And so this ends up being fairly high dimensional data that can be difficult to visualize and sort of abstract mentally. If we change how we think about it, though, it's easy to compare it to something that we're all familiar with. So if instead of beta diversity or genetic dissimilarity, we think of the beta diversity score between each pair of samples to just be a distance. So essentially, taxa that are samples that are more distant from each other are more dissimilar, while taxa that are closer together are more similar to each other. We can then kind of compare that to something we're very familiar with, which is maps and geographic maps. So if we think about a map, we can represent the distance between any 2 points on a map as a two-dimensional projection. We know that in real life, the Earth is a sphere in 3 dimensions. So that's actually a more difficult distance to determine, right? So we have to account for the curvature of the Earth and changes in altitude. And so it's not necessarily a two-dimensional distance that we're actually looking at. But we can project that 3-dimensional distance on 2 dimensions with maps that we're all familiar with. Now, we all know that there's no perfect projection of the Earth. It's impossible to project a 3-dimensional globe onto 2 dimensions. But the one thing maps typically do pretty well is conserve the distance between any 2 points. And that's what we see here. And we can do something very similar with our high-dimensional beta diversity data using a tool called principal coordinate analysis, or PCOA. What PCOA does is it essentially takes our very high-dimensional beta diversity data and finds a projection of that in either 2 or 3 dimensions that maintains those relationships in terms of distance from distance between any 2 points. So by reducing the dimensions, we can find a consistent representation of our data. One side effect of this is that there may be many different projections that accomplish this, and often when you rerun PCOA, you might see different orientations of essentially the same data. But what this allows us to do is to visualize our data and ask some questions about how these data cluster and are located in relative to each other. So for instance, we can take a look at this PCOA projection of our beta diversity metrics and ask if, maybe they have different centroids. So clustering between different groups of samples, maybe healthy and unhealthy samples, clusters around different centroids. Or if we want to ask more statistically and to determine if there's a significant separation between different sets of samples, we can use a tool called PermaNova. The PermaNova sounds a little bit confusing, but it's actually just a variation of a statistical test that many of us are probably familiar with, which is an analysis of variance test, or ANOVA. ANOVA essentially seeks to answer whether changes in an independent variable can account for the differences in the variance of a dependent variable. So essentially, if you add salts to a glass of water, how does that account for variances in the final pH of the water? Very likely it does. So that works great if we have one dependent variable, but for our beta diversity, we actually have several hundred dependent variables, which is the diversity of all of our taxa in our samples. So for something like that, we might use what's called a MANOVA, or a multivariate analysis of variance test. And so this works pretty well if we have multiple dependent variables, and we could use this, except that our beta diversity data is very heterogeneous and not normally distributed. And so we might run into some issues if we use a classic MANOVA statistical test. So instead, we use what's called a permuted multivariate analysis of variance, or PermaNova for short, since that's quite a mouthful. So PermaNova essentially works by taking our real regression model, wherein we group our samples based on what group they cluster with. So in this case, maybe healthy individuals versus individuals with recurrent C. difficile infection. We run that regression model and we return what's called an F value, which essentially measures the goodness of fit, how well our regression fits the data. We then scramble or permute those regression models. So essentially randomly assign samples to different clusters. And each time, we do this several thousand times, and each time we again fit the regression model and calculate an F value that determines how well that regression model fits the data. We can then compare the F value of our real regression model that we ran initially against all of these scrambled or permuted F values. And this comparison actually returns a p-value that will allow us to determine if the real regression model is statistically significant in terms of describing the variance in our data. We can also calculate the overall amount of variance that's described by that regression model. We'll actually go through doing exactly this in the notebook here shortly when we switch back over. So actually, let's right now go ahead and do that. At this point, the denoising step in data 2 should have been finished. So let's switch back over to the notebook and take a look. Yep, so it looks like that denoising step took about 6 minutes for me. Hopefully yours is done at this point as well. If you ran into any issues, please don't hesitate to ask the TAs. They'll be happy to help you. And so, excuse me. If you weren't able to run that step, you can run this cell underneath, which will essentially copy over all of the results from the data process into your current directory from that treasure chest where all of our outputs are stored. So what we can do now is actually take a look at how that denoising step worked and visualize the statistics that come out of that. So actually, before we do that, let's take a look at the output that we got from the data 2 process in our file directory over here. So we now can see our data file directory that's within that materials directory. And we see that there's 3 new CHIME artifacts. One is called table, and that just contains the abundance of each of our identified ASVs. The second one above that is called representative sequences, and this contains the actual ASVs that were identified by the data 2 process. Lastly is this thing called denoising stats, which is essentially a summary of how well that data 2 step worked and what sort of filtering it did. So we might be interested in taking a look at this. And so let's go ahead and run this cell, which will use the metadata plugin to tabulate our denoising stats into a visualization. And then we can go ahead and switch back over to the CHIME2 viewer to take a look at the results of that denoising step and see how successful that was. This really shouldn't take very long, but it will be very informative in terms of telling us how many of our sequences were filtered out and whether we have enough data to continue moving on. Great, so now within this data directory, we can see the denoising stats.qzv file. We'll go ahead and download that and switch back over to our CHIME2 viewer. So let's close this. If you click on this header up here, this will take you back to this homepage. Here we can upload our second visualization, which is our denoising stats. So again, here we just have essentially a data table that describes the results of this step. On the left, we can see all 8 of our samples are listed. And this first column just tells us how many reads we had in each one of these to begin with. So again, we have between 25,000 and 125,000 reads to begin with. The second column will tell us how many of those were filtered out by that initial filter and trim step. So that will remove any reads that are just of low average quality. And we can see here that for all of these, we have, with the exception of one, we have upwards of 98% of reads passing the filter. And this one was still upwards of 90%. And so that tells us that our sequencing quality was actually very good. We shouldn't be too worried about the results that came out of our sequencing. Data 2 then, so this will also list samples that remain after our denoising. So this is essentially removing any reads that couldn't be assigned to an ASV within data 2. It'll also then tell us how many of our reads were non-chimeric. And so after removing chimeras, how many reads were left. And we can see in this last column sort of how many of our initial reads passed all the way through to the very end of this data 2 step. And for most of them, we see that it's upwards of 80% of our reads are still remaining. One of them seems to be a little bit lower, but for the purposes of our data today, of our process today, that'll be just fine. And we don't have to worry about removing data or anything. Okay, so let's go back to the notebook and start taking a look at diversity. So quick overview again, we're looking at alpha diversity within samples as well as beta diversity between samples. And the first thing that we have to do, like we mentioned, is build our phylogenetic tree. So again, using our representative sequences, we can use the phylogeny plugin within CHIME to build this phylogenetic tree. So let's go ahead and run this cell. And what this will do, it will take our representative sequences and build several CHIME artifacts and output them to a directory that we'll just call tree. And we'll take a look here in a second at what exactly that does. But what we'll also do is actually have a look at our tree and see what that looks like. So for visualizing our tree, we can use what's called the Empress plugin within CHIME2. And so what that will do is we'll take the CHIME artifact that we just constructed. We see our tree directory on the left-hand side here. We now have 4 new CHIME artifacts, including one called rooted tree. So rooted tree is what we'll essentially be using moving forward as our phylogenetic tree. And if we run this cell, CHIME will actually build us a visualization of our tree that we can again take over to the CHIME2 viewer, take a look at what that tree looks like and see what we can kind of glean from that. So let's go ahead and do that. We can go to our file directory again, download this new visualization that's called empress.qzv, and bring that over to the CHIME2 viewer. Again, if we click on this header, that'll take us to the homepage again, where we can upload our newly constructed phylogenetic tree. So right now, it doesn't really look like much. And without knowing the specific taxonomy assignments of each of our ASVs, we won't really be able to learn a lot from this tree. Right now, we can change though the layout that we choose to view our tree. Right now, it's in what's called an unrooted format, which essentially means there's no sort of central root from which all of the sequences begin. We can also switch that to be a rectangular view, which you might be more familiar with, or a circular view. So now we do have sort of a rooted tree. So this is the sort of root sequence. And we can see that there's immediately some divergence into different clades. If we want to change the branch lengths, right now, the branch lengths are just considered normal, which means that branch length correlates to genetic dissimilarity from each other. So longer branch lengths mean more genetic divergence between clades. We can choose to ignore branch lengths and make it all very similar, or we can make it ultrametric, which essentially just means that branch lengths are adjusted so we get this nice circular format. So like I said, there's not much we can actually get from this phylogenetic tree right now. But later on, at the end of the course, there'll be a task for you to work on your own, where you'll actually return to this phylogenetic tree and label it with some associated metadata, as well as our taxonomy assignments, to learn a little bit more about our communities. Okay, so let's go back to our notebook for now, and continue on with calculating diversity metrics for our communities. So, as you might expect, CHIME actually has a plugin called Diversity, which we can use to calculate many of the diversity metrics we're interested in today. We can call that CHIME plugin, and specifically the action called Core Metrics Phylogenetic, which will then use our table of ASV reads that we got from data 2, as well as the tree we just constructed in the last step, to calculate several different diversity metrics that we might be interested in. One of the other things we have to add to this step is what's called sampling depth. And this is actually to normalize the data due to something that is actually an artifact of sequencing. So if you recall, when we were looking at the sequence reads from each of our samples, we saw some pretty drastic variance in the number of reads. One of our samples had 125,000 reads, while another had far less, I think about 25,000. And so you might think that that's actually a function of, the number of taxa in a sample or the amount of DNA that's present in a read, but that's actually not the case, since it's actually standard practice to normalize the amount of DNA going into each sequencing sample. So the number of reads is actually just a sort of an artifact of random chance in sequencing, but it can drastically affect our results if we don't do anything about it before calculating diversity. And that's because of this. If you imagine a sample with 500,000 reads, any taxa present in that sample that are super rare, that don't show up a lot, are more likely to be sampled in that sample than, for instance, a sample wherein you only have a couple thousand reads. So if we calculated directly the alpha diversity of those two samples, we would artificially see that there's actually higher alpha diversity in the sample that had more reads than in the one that had fewer reads. So to combat that, what we do is we identify the sampling depth, which essentially tells CHIME to take 8,000, to randomly select 8,000 sequences and use only those sequences to calculate diversity. It's not a perfect system, since we end up losing a lot of our data, but it does mean that we can compare samples with different sampling depths directly to each other. We'll also add in this metadata file, which we'll use later on when we want to compare the diversity metrics between different groups of samples, and identify where we want our diversity artifacts to be saved, which is this diversity directory. So CHIME will now go through and calculate alpha and beta diversity metrics for all of our samples using these data, including the phylogenetic data that we got from our tree that we just constructed. And in just a second, we'll use these to take a look at both alpha and beta diversity. Great, so that seems to have worked pretty well. It took about 20 seconds on my end. And so if we refresh our directory over here, we'll again see a brand new directory, a brand new file folder called diversity. And if we open that up, we can see all sorts of new CHIME artifacts as well as visualizations. We could download any of these visualizations and take them to the viewer to get an idea. But first, what we'll actually do is take a look at Shannon index, which, if you'll recall, is the metric we'll use to estimate alpha diversity between our samples. In this case, CHIME didn't create a visualization for our Shannon index, so we'll have to do that ourselves. And so in this step, we'll again call the CHIME diversity plugin, and in this case, use the action alpha group significance to build a visualization of our Shannon index. So we can run this cell while we take a look at exactly what we did. As an input file, we use that Shannon vector we just calculated. We also include the metadata file, and you'll see why that's important in a second, since really what we're doing is comparing the alpha diversity between our healthy and unhealthy groups. Okay, so now it's saved this visualization to our diversity directory in a CHIME visualization called alpha groups. So if we go over here, same process as usual, download the alpha groups visualization, go back to the CHIME2 viewer, start over here, can take a look at our alpha diversity plots. So here we can see on the left-hand side, we have our Shannon index. Right now, the default x-axis is the collection date when samples were taken, and that's not very informative since we have mostly one sample per collection timestamp. So if we go up to this drop down at the top that says column, we can select which metadata column we want to use as our grouping. In this case, we'll click on disease state, which will separate it into healthy and unhealthy individuals. So now we can see a comparison of the alpha diversity for both healthy individuals on the right-hand side, as well as individuals with recurrent C. difficile infection on the left-hand side. As you can see, there's a pretty drastic decrease in overall alpha diversity in those folks with recurrent C. difficile infection. Generally, in longer living, very healthy individuals, we see higher alpha diversity, although it's not a perfect metric of health or unhealth. But this generally agrees with sort of the knowledge in the field that is having really low alpha diversity can be a general indicator of unhealthiness. If we scroll down, we'll also see that CHIME went ahead and did some significance testing for us. So in this case, it ran a Kruskal-Wallis test, which is a non-parametric significance test, to see if there is a statistically significant separation in alpha diversity between our two groups. And in this case, we see we have calculated an H value as from our Kruskal-Wallis test, as well as a p-value that tells us that this is, in fact, a statistically significant result. The numbers in your visualization might vary a little bit from what you see here, and that's just because we randomly selected 8,000 sequences from each sample. So the sequences in your alpha diversity metrics might be a little bit different from mine, and that can change the results a little bit here and there. Cool, so that's essentially an overview of alpha diversity. It gives us a good idea of how we can measure within sample diversity between all of our samples. So let's go back to our notebook now, and with the last few minutes, we'll take a look at beta diversity before we take a break. So for beta diversity, in this case, we will take a look at the weighted unifrac distance. So if you recall, weighted unifrac is a measure of beta diversity that takes into account phylogenetic relatedness, as well as abundance of taxa within our samples. So before we check for significance, let's go to our file directory and download the weighted unifrac _ emperor file. So emperor is another plugin that allows us to visualize these things. And we'll download this visualization and take a look at it as well. Okay, so now that we've downloaded it, we can switch back over to the viewer and open up this beta diversity visualization. So if you recall, what we'll see here is a PCOA projection of our beta diversity. So again, beta diversity is very high dimensional data that tells us essentially the distance between each of our samples. And in this case, we see the PCOA projection first in 2 dimensions, but we can also click and drag and see that across 3 dimensions. The axes on each of the axes also contains a label that describes the amount of variance that can be described by that axis. So we can see axis 1 describes about 2/3 of the variation that we see in our data already. And if we combine that with axis 2, we're up to nearly 90% of the overall variance in our data. So that's pretty good. It looks like we found a projection that can really describe the beta diversity of our data pretty well. Right now, though, since we just have a bunch of red dots, that doesn't mean very much to us. What we can do, though, is go to this right-hand side to that first dropdown. It says select a color category. And again, choose that disease state column from our metadata file. And this will color the dots accordingly. Great, so now we can see the beta diversity between our healthy samples in red and our pre-FMT or unhealthy samples in blue. And we can actually see that, especially along axis 1, these diverge pretty significantly. So it looks like healthy individuals are more closely, are closer to each other, sort of on the right hand of the axis, while unhealthy individuals are closer to each other on the left hand of the axis. And if we rotate the projection, we can see that they might also cluster around different groups. And this might be due to a few different reasons, right? So we can take a look from the top down. There's also a little bit of clustering that happens with some of the healthy individuals. And if we had additional metadata files, or metadata, if we had additional metadata for all of our samples, we could color by that as well to determine if there was any significant clustering going on. But if we want to actually measure statistically whether the separation between these samples is significant, we can go back to the notebook and conduct our permanova that we were discussing earlier. Okay, so the last piece for our sort of diversity metrics here is doing exactly that. So using this unifrac distance matrix that we constructed in that core metrics phylogenetic step a few minutes ago, we can input that into this Adonis action, which will go through and actually conduct the permanova that we described in the slides. We pair that with our metadata file, as well as the specific column in the metadata file that we want to use as our sort of clustering. So in this case, we're testing for significance between healthy and unhealthy individuals. So if we go ahead and run this, now CHIME will go through and calculate that permanova and tell us whether or not this is a statistically significant separation in terms of beta diversity between our healthy and unhealthy samples. Again, this will output a visualization into our diversity folder that we'll call permanova.qzb. And this will be the last thing that we do here for now. But we can download this file on the left-hand side. And one last time, head back over to the CHIME2 viewer, click and drag, and open up our permanova.qzb visualization. So this is just a data table that describes to us the results of the permanova. In this case, we were looking at if the separation between healthy and unhealthy individuals was significant. And if we go all the way over to the right side, we see the sort of the calculated F value for our model. And we also see this R squared, which describes the percent of variance that can be described by our regression model. And so that's about 56% of the variation that can be described just by clustering between healthy and unhealthy individuals. And that's pretty significant. So that tells us that this is a pretty significant result. And indeed, the p-value is 0.029. So anything less than 0.05 we consider to be statistically significant. So that tells us this result is in fact significant, and there's definitely a separation in terms of beta diversity between healthy and unhealthy samples in our data set. Great, so at this point, we'll switch back over to the notebook very briefly, and we'll have a little teaser for what we're doing next. So sort of the last piece of this CHIME2 pipeline is identifying what organisms our ASVs actually are. So right now we have these representative sequences, and we have sort of the sequences from the V4 region of the 16S gene, but we don't know what taxa they each belong to. So maybe during the break, you can think about how you would go about going from a sequence directly to an organism's name, sort of assigning the identity of these taxa to their sequences. But with that, I think we're out of time for this part of the session. And so we'll take a break here, and we'll be back in 20 minutes, I believe, to finish up the course. So go and take a little break, get a glass of water, and I'll see you back here in about 20 minutes. Also happy to take any questions right now if there are any that are outstanding on Slack. But if not, then yeah, feel free to ask questions during the intermission as well. The TAs will be happy to help you there. Thank you very much, and I'll see you in 20 minutes. All right. Welcome back, everyone. I hope you had a nice little break. But welcome back to the course as we continue on here. So if you recall, up to this point, we've started with our raw sequencing reads. We've gone through importing our sequencing reads into CHIME 2, denoising them using data 2, wherein we identified our ASVs. We built a phylogenetic tree using our sequences, and then use that phylogenetic tree to calculate and visualize some of our diversity metrics, both alpha and beta diversity. The last step, and one that I think is pretty exciting, is sort of what I teased at the end of the last part of the last session of the course, which is identifying exactly which taxa are present in our samples. So as a result of that ASV identification process within data 2, we now have a list of representative sequences. Essentially, these are sequences that represent the original sequences present in each of our samples. Right now, they don't have a whole lot of meaning since they're just really long strings of bases. And so what we're interested in is going from those sequences directly to the identity of the taxa from which that came. And this can help us to identify the composition of someone's microbiome and use that to give more context to what to how their microbiome might be affecting them, right? So in terms of how at risk they might be for infection of pathogenic bacteria, such as C. difficile. So before the break, I asked you to kind of think about how would you do that? How would you go from a representative sequence, one of our ASVs, to the identity of an organism? But before we really answer that question, let's quickly consider and have a little refresher on taxonomy and how that sort of works. So I think we're mostly all familiar with taxonomic ranks, but essentially we have these hierarchical ranks which get more and more specific. So down at the species level, we have the most sort of specific taxonomic rank, wherein there's very little genetic diversity at that level. At each level that we go up from the species level, we see a little bit more diversity, both like a little bit more genetic diversity and divergence between species. So at the genus level, for instance, there's more diversity than at the species level. For bacteria, this can be a little bit challenging, and we can actually get even more granular down to the strain level, which is essentially any given genome. But this is generally how it's done. An important thing to note is that with the 16S data that we're using for today's course, the lowest taxonomic rank that we can confidently assign to our reads is the genus level. And the reason for that is below the genus level, at the species or strain level, there's simply not enough diversity in that 16S gene to confidently say it came from one species versus another. So we'll be doing all of our taxonomic assignments to the genus level. We run into an issue here, though, because like we mentioned, there is some genetic divergence and variability within that genus level. And so this can actually bring up an issue with what I think is probably the most intuitive approach to the question that I posed on that last slide. So you might think, we have this list of representative sequences, or ASVs. We could just compare that directly against a database of known genes, and using that, look for matches and determine exactly where our ASVs came from. But this doesn't always really work well in practice. And the reason for that is that the database that we'll use to compare, the ASVs against our reference isn't always representative of the actual taxa in our samples. Some taxa might be missing from the database, and still others that might be, the taxon might be present. The strain that was used to generate a reference sequence might diverge a little bit genetically from the strain that we actually find in someone's gut microbiome. So for instance, it might be a lab strain of E. coli that was used to generate a reference sequence in our database, but that's a little bit distinct from the actual E. coli we see in someone's microbiome. So instead, we need to think of sort of a more general approach, more generalizable approach that allows us to determine whether or not the sequence that we're looking at in our ASVs generally matches what's present in our reference database. So one thing that we could consider is codon usage. So we know that the DNA code is degenerative. So that means that there's multiple codons that can code for the same amino acid. And in practice, sometimes the specific codon usage can be taxon specific. So some taxa will use one codon versus another for the same amino acid. So this is helpful, but it doesn't really mean that we can use that to identify all the different possible genera that are present in our samples. But it does give us an idea for how we can approach this problem. So instead of looking for exact sequence matches, what we can consider instead is whether the overall sequence is generally conserved between the taxa that we see in our samples and our reference database. And so to do exactly that, we use what's called a multinomial naive Bayes method. And so this sounds a little complicated, but really it's exactly what I was just describing, and just involves a little clever math in order to get to our classifications. So let's say we begin with a query sequence. In this case, we see the query sequence on the top left here. It's, as an example, it's just 6 bases long. To begin, we can subdivide the query sequence into all possible subsequences that are shorter than that initial query sequence. And these are commonly referred to as k-mers. So in this case, we can divide this query sequence into these 3 base long subsequences. So we have an ACG, a CGC, a GCG, and another CGC. And these k-mers are just subsequences of length k. So in this case, we've constructed 3 mers. And it turns out that if we subdivide our initial query sequence into all possible k-mers, we can build a profile of the frequency or probability of finding these k-mers within that query sequence that can be relatively taxon specific. And this essentially allows us to determine exactly what we were discussing earlier, which is, does the composition of this sequence stay somewhat conserved between the sequence we see in our data and our reference model, while allowing for some genetic divergence between our query sequence and the reference model. And this accounts for the fact that generally, through evolution, as the sequence might change here and there, it won't do so drastically. It'll mostly be small pieces here and there. And so the overall composition of that sequence will remain mostly unchanged. So let's take a look at our reference model on the right-hand side here. So in this case, our reference database, as an example, we just have 2 taxa, taxon 1 and taxon 2. The first thing we might consider is the probability of finding one of these taxa within our reference database. So for instance, if our reference database has 15 different strains of E. coli that are represented, but only 1 strain of Faecalibacterium, for instance, we might consider the probability of selecting one of those E. coli strains over the single Faecalibacterium strain. This is called the prior probability. And this is then different methods differ on whether or not they consider that or not, but it's an important thing to take into account. As we're building up our reference model, essentially what we do is we take our reference sequence for each of our taxa, and we calculate the frequency or the probability of finding each of these k-mers. So for instance, for taxon 1, the probability of finding an ACG is about 25%. Probability of finding a CGC is also 25%, and so on. And so we can do this for all the taxa in our reference database and compile a profile for each of these k-mer frequencies. In and of itself, these k-mer frequencies are not that useful. And that's because essentially they say, what is the probability of finding a query given a specific taxon? And really what we're interested in looking at is the opposite question. What is the probability of finding a taxon given a specific query? And this is where Bayes method comes in. So in multinomial naive Bayes, this is the Bayes part. So Bayes' theorem is shown down here on the bottom right-hand side. And this allows us to sort of flip that probability around. So using some clever math, we can go from that probability of finding a query given a specific taxon to finding the probability of finding a taxon given a specific query. And to do that, we can essentially go through our query sequence and all the specific k-mers of a query sequence and find the corresponding probability of finding each of those k-mers within each taxon in our reference model. So let's take a look at this top line first. First, we take the prior probability. So again, the probability of finding this taxon 1 in our reference database, and multiply that against the probability of finding each of our specific k-mers from our query sequence in taxon 1. So we have 1 ACG, 2 CGCs, and 1 GCG. We find those corresponding probabilities, multiply them together, and we're left with the probability of finding taxon 1 given our query sequence. We can do the same thing for all the different taxa in our reference database, and we'll be left with one taxon that will have the highest probability of being present given our query sequence. In this case, that's taxon 1. One thing you might notice is that in Bayes' method here, we are ignoring the denominator down here, which is the probability of finding a given query. In practice, though, we essentially assume that finding any specific query within our sample data has the exact same probability. And so we can effectively ignore that without changing the functional probabilities of finding a taxon given a specific query. So in the end, we just choose the taxon with the highest probability and assign that query sequence the name of that taxon. In practice, you can really use any sort of machine learning method that's based on k-mers to do this. But we will use, today we'll use multinomial naive Bayes. And it turns out this is actually a pretty robust and accurate method that gives better generalization than just aligning sequence reads directly. And it also gives much faster results. So this is a pretty quick process overall. So now that we've kind of discussed how that works, let's switch back over to the notebook and begin assigning taxonomy to each of our ASVs. Great. So hopefully you still have the notebook open and hopefully it's still connected. If you've run into any issues where your notebook has disconnected for some reason or you've lost any of your data, don't hesitate to reach out to the TAs. As a reminder, any of the outputs that we've already produced to this point are available to you in that treasure chest directory within that materials folder. So if you have to restart, you can always find the CHIME inputs that we'll be using here in that treasure chest folder. So let's take a look at how we can use CHIME to classify the taxonomy of our ASVs. The first thing we'll do is use a plugin in CHIME called Feature Classifier. So as you might guess, this will take our ASVs and do that multinomial naive Bayes method that we just discussed in order to assign them taxonomy. Specifically, we'll be using the classify-sklearn action within that plugin. As inputs for this function, we'll be using our representative sequences. So again, this is just our list of ASVs as identified by data 2 earlier in the course. We'll also give it a classifier. So in this case, this is a specific classifier that contains all those k-mer profiles for several reference taxa. And this one is actually collapsed to the genus level. So if you recall, we'll be doing all of our classification to the genus level, since that's the most granular taxonomic level to which we can confidently assign taxonomy. And you'll also see this last bit here that tells us that this classifier was actually trained specifically on the V4 region of that 16S gene. So this is very specific to the data that we're working on today and should return us very specific results, as opposed to using a classifier that's trained on the entire 16S gene. We'll again assign 2 CPUs to get this job done and go ahead and run this cell. As an output, we'll end up with a new CHIME artifact called taxa.qza. And this artifact will contain all of the taxonomic assignments of our ASVs that we had stored in our representative sequences artifact previously. So this can take a couple minutes here, really shouldn't take too long. But now we can kind of start to consider what we will do now that we have our ASVs classified for taxonomy. The first thing that we'll consider is maybe visualizing our data in terms of the taxonomic breakdown of each of our samples. So what we can do, in fact, is use another plugin within CHIME called Taxa, which will take our list of abundances that we produce from data as well as our taxonomic assignments that are currently being calculated, and use those to create a visualization for each of our samples in terms of their taxonomic composition. And so in this case, we're also adding the metadata file so that we can have some associated context for each of our samples. Let's go ahead and run this next cell, which will take those classified reads and visualize them as a taxonomic breakdown of each one of our samples. And for this, we'll again, once this cell finishes, we will download the output and bring it over to the CHIME2 viewer like we did in the first part of the course, and take a look at what we can find and what sort of insights this might give us into our data. Okay, so if we scroll all the way to the bottom of our, if we scroll all the way to the bottom of our file directory, we will download our taxa_barplot.qzv. And so now that's downloaded, we'll go back over to the CHIME2 viewer and take a look at what this visualization looks like. Okay, so hopefully yours looks like this as well. So right now it's a little hard to see, everything's a little bit cramped. We can use this slider on the top left to increase the width of our plot, just to make everything a little bit easier to see. Okay, so now we see a bunch of green lines, and that doesn't really tell us very much. But what's happening here is that CHIME will automatically give us this summary at the highest taxonomic level. So right now, all we can see is that all of our reads are classified as bacteria, which makes sense. That's generally what we expect to see here. But what we can do is use this first dropdown to specify the taxonomic level that we're interested in. If we go to level 2, this is the phylum level. And now we can see that there's a distinct composition for each one of our samples. If we go even more distinct, we can go down to the genus level, which is level 6. And now we can see that for each of our samples, we have, dozens, if not hundreds of different genera that are present in each of these samples. So this is cool, but without additional context, we can't really make any insights into this, right? But what we can do is go to the third dropdown over here that says sort samples by. Click on that and select disease state. So again, this will separate our samples into healthy individuals and individuals with recurrent C. difficile infections. So let's click that. And now we can see the healthy individuals on the left-hand side and those folks that have recurrent C. difficile infections on the right-hand side. And now this is pretty neat, right? So we definitely see a signature that looks to be related to healthy individuals, which looks like a little bit more abundance, as well as even, higher abundance of, higher richness of taxa. So more taxa seem to be present, as well as more evenness of taxa, right? So, and this fits with what we had seen in our diversity metrics, where in the alpha diversity of our healthy individuals was much higher, significantly higher than the individuals with recurrent C. difficile infection. And if we look at those individuals with the infection, we can see that they're generally dominated by a couple taxa and look to have a little bit less species richness within those samples. So I encourage you to, take a look at this, see what else you can maybe determine about the microbial composition of our samples in the future. We can also sort by other sample metadata if we wanted to, and also look at other taxonomic levels that might interest us. You'll also notice that if we scroll over any part of the bar plot, we can see the associated taxonomic assignment at the top of the bar plot, as well as the % relative abundance of that taxon within a sample. Great, but for now, let's switch back over to the notebook and continue with our analysis. So now we have our taxonomic assignments. We also have our abundances of each of our ASVs. One thing that we might be interested in doing is collapsing those together at the genus level to build a table that essentially has the abundance of each one of our assigned taxa for each of our samples. And that's what this cell will do. We'll use that taxa plugin and the collapse functionality within it to take our table containing our abundances, as well as our taxonomic assignment, and tell it to collapse to the 6th taxonomic level, which again is the genus level. And this will save a new CHIME artifact that we'll call genus.qza. Now, if we go to our file directory, we can see that on the left-hand side. You might have to refresh your directory again, and there it is. One thing that we might be interested in now that we're sort of reaching the terminus of our CHIME2 pipeline is how do we export our data that's currently as a CHIME artifact into a form that we can use for other tools. Say maybe we have additional computational pipelines, or we want to bring our data out and do some analysis in R. We might need the data in a different format. And to do that, we can actually do the opposite of what we did at the beginning of the course, which is use this CHIME tools export functionality. So if we run this cell, essentially this is taking our input path, which currently is our genus.qza artifact that we just constructed a moment ago, and defining an export path. So what this will do is take our genus.qza artifact and save it, export it, and save it to a directory that we'll call exported. If we look at that exported directory, what we see is something called featuretable.biome. So .biome is another data type that's useful for some analyses, but it's not the most easily readable for us. So to the cell, we also added a second action called biome convert. So what this will do is actually call the biome library, not CHIME, and convert that biome file that we just exported to be a tab separated value table. So here we're taking featuretable.biome and creating something called genus.tsv. And that tab separated value file will be much easier for us to read later on in the course. So let's take a look at our file directory. And there it is. There is genus.tsv. So now it's probably a good idea to take a look at what exactly this table that we just constructed actually looks like. So let's run this cell, which again will use pandas, which is our data frame management tool, to read this tab separated value file and print it out for us. Great. So this is what it looks like. And what we can see is a couple things on the index of this data frame. So that farthest left column contains our taxonomic identifiers. So we've assigned taxonomy to each of our ASVs, and this is the that corresponding taxonomic assignment. You can see that it covers the entire sort of taxonomic hierarchy. So starting at the kingdom level, all the way down to the genus level. You'll also notice that some of these aren't annotated all the way to the genus level. And that's because our multinomial naive Bayes methodology can sometimes determine taxonomy down to higher orders of taxonomy, but not necessarily all the way to the genus level for some reads. So for those, you might see a couple of sort of semicolons followed by underscores with no additional data. To the right of this data frame, we'll also see a column representing each one of our samples. So again, we have 8 samples, 8 columns, and the values within each cell represent the count of reads that correspond to that taxon within that sample. So this data format is pretty useful actually, because we can see sort of the abundance of each taxon within each sample. And tomorrow, Alex will take you through a process of using data that looks a lot like this to build mechanistic models of the gut microbiome. So you can see how this would be pretty useful information. But for now, what we'll do is actually, now that we've exported our data out of CHIME, it's actually useful to us in terms of applying it to additional packages or visualization tools. So if we're more familiar using something like Seaborn or Plot9, now that we've exported our data out of CHIME, we can visualize our results using one of those. And so that's what we'll actually do down here in this block of code. Is we will build a visualization, specifically a heat map, using a tool called Seaborn. Before we start doing that, we need to import 2 more packages into our notebook environment. First, we'll import something called NumPy, which will allow us to do some mathematical analysis and methods. And then we'll also import Seaborn, which is our visualization tool, into our environment. Before we build our visualization, we'll need to transform our data a little bit and format our data frame to be a little bit more appealing visually. The first thing we'll do in this first line of code is sort of clean up the ASV identifiers that show up in this first column of our data frame. So like we can see, we have each of these identifiers covers the entire hierarchy of taxonomy. But that might get a little bit clunky on our heat map. So instead of doing that, we'll use this string split functionality to split each one of these super long strings into a bunch of substrings. And it'll do that every time it sees the semicolon, since we identify the semicolon as our operator. Then we'll tell it to select only the sixth string. So since we start counting from 0, string 5 is actually the sixth string, and save just that string. So what this will do is drop all of those substrings prior to the final substring in our index, which is the genus identifier. And that'll make our plot just a little bit more easy to read and understandable. We'll then also tell it to drop any rows where the genus isn't identified. So if we see something like this, where we just have an empty identifier where it should be identifying a genus, we'll tell pandas to drop those rows. Lastly, since we have a pretty big data frame, this is 163 rows total, we'll tell it to take a random sample of 50 rows from that data frame. So 50 random genera from our abundance data frame that we'll use for building our visualization. Okay, now last thing before we build our visualization is we're going to do another normalization technique. So this time we'll do what's called a center log ratio transform. So that sounds a little confusing, but essentially what we're doing is for each data point, we take the log of its abundance and subtract that from the log of the mean of the abundance. And essentially what this does is it allows us to take this data, which as you can see sort of spans several orders of magnitude from really large numbers of breeds down to just 1 or 2. It allows us to compare those more directly. And a way to understand the center log ratio a little bit more easily is a value of 0 for a center log ratio indicates that that specific taxon is as abundant as the mean. A positive value means it's more abundant than the mean, and a negative value means it's less abundant than the mean. So this is just a way for us to normalize our data in a way that makes it more understandable and more easy to compare on a per sample basis. And then finally, the last thing that we'll do is actually call Seaborn, our visualization tool, and build what's called a cluster map. So this is essentially a clustered heat map that will take our new data table, just called transformed after our center log ratio transform, and we'll actually take the transpose of that. So we'll just flip it on its side, essentially. The last few things are just formatting. This will be the color map that we use to color in our heat map. We'll turn on tick labels, which you'll see in a second, and then also define the figure size. So if we go ahead and run this cell, this should only take a second or two, and we're left with a pretty nice looking heat map. So you can see on the x-axis, we have just our genus identifiers for those genera that are identified. We have a random sample of 50. So keep in mind that since we all sampled 50 random genera, your heat map might look a little bit different from mine. Each row represents one of the samples in our data. And then the coloring of each cell represents the centered log ratio transform of the abundance of a given genus within a sample. And so you might start to notice some interesting patterns here. For instance, Villionella seems to be really abundant in these first three samples, and less so in the last 5 or so. You might also see some taxa that are highly present in one sample, like here, and not so much in others. You might see some taxa that are present across a lot of samples, but missing from a few others as well. So this is just one way that we can look at our compositional data in a way that will allow us to infer a little bit more about our community structure and learn a little bit about the composition without getting too in the weeds. So I encourage you to take a look a little bit closer at this, maybe sample 50 other random genera, and take a look at how the output changes. If you were really interested, you could also append the metadata that we looked at previously to determine how some of these compositional differences might vary between healthy and unhealthy individuals. And if you find anything cool, make sure to put it on Slack, and we'll be happy to listen to maybe what your findings look like. So yeah, that's sort of how we can use CHIME data, export it out as a more readable file, and then use that in any other visualization tool that you might be more comfortable with. All right, so that's the end of the data analysis portion of our course. At the end of this notebook, you'll find an extra exercise that I'll leave for you to work on your own, wherein we'll revisit that phylogenetic tree that we constructed earlier in the course. Now that we have our taxonomy assignments, we can actually add to that some metadata in terms of which samples come from healthy and unhealthy individuals, as well as the taxonomic assignments of each of our taxa. And that will construct a new visualization, which you can take over to the CHIME 2 viewer and answer some of the questions here. Again, feel free to share with us any cool things that you might learn from this visualization. You'll be able to annotate that tree based on healthy and unhealthy individuals and see if there's any clustering of taxa between the two groups. So on your own time, maybe before the next talk, take a look at this exercise. And if you have any questions about it, feel free to ask the TAs on Slack. We'll be happy to help you. But for now, let's switch back over to the slides one last time. So yeah, like I said, now it's your turn to do some coding. Go ahead and take a crack at that exercise. And feel free to use that Colab notebook to play around with CHIME some more. Maybe you have some ideas for other analyses or investigations that you can get out of our the data that we analyzed today. And we'd love to hear about it. So with that, we are done with today's portion of the course. Before we end, I'd like to thank everyone that was involved with making this happen, including our instructors and our wonderful TAs that have been following along and answering all your questions on Slack, as well as all the people behind the scenes that have helped to make this course possible. Thank you all so much for being here today. I do hope you'll stick around. In about an hour or so, we have a wonderful talk by Katherine Ramos, who will come in and tell us a little bit more, tell us about her work determining some of the factors that raise or lower someone's risk for C. difficile infection. And that'll be really fascinating. So I really do hope you'll join us for that. And then also that we'll see you at tomorrow's part of the course, where Alex will take you through some mechanistic modeling, wherein we can model the invasion of C. difficile into the gut microbiome and determine which individuals are more at risk than others. So with that, thank you so much for being here. We'll now have a short break until Catherine's talk at 12:45. Thank you. So we have just under an hour until that talk. So go ahead, take a break, and we'll see you back here at 12:45. So just under an hour from now. Thank you so much. Hello, everyone, and thank you for joining us here. I hope you all enjoyed this morning's portion of the course and that hopefully you learned something. Been getting a lot of great questions coming in via Slack, so please don't hesitate to ask some more as they come up. Coming up right now, we'll have another very talented member of the Gibbons Lab, Katherine Ramos Sarmiento, joining us to give a talk on some work that she's been doing, applying island biogeography theory to the human gut microbiome. I encourage you to listen and learn something from her as well. During this portion, we please ask that if you have any questions, you ask them via the Q&A function on Zoom. You can also vote on other people's questions there. And the top vote getting questions at the end of the talk will be the ones that we choose to answer. So with that, thank you for your attention. And Katherine, the floor is yours.
Hi, everybody. Let me just share my screen. Okay. Okay, hi. Oh, wait, this is the wrong slide. Okay. Hi, everybody. My name is Katherine Ramos Sarmiento. I am a research associate in the Gibbons Lab, and today I'll be talking to you about some of the research I've been doing here, titled Island Biogeography Theory and the Gut: Why Taller People Tend to Harbor More Diverse Gut Microbiomes. So, some background on the human gut microbiome. The microbiome refers to the cumulative genomes of the microbes in the environment, and the microbiota refers to the microorganisms that exist in the environment. But a lot of people use them interchangeably, and that is what I will be doing today. There are 38 trillion bacterial cells in and on our bodies, which is particularly impressive when we consider that there are about 30 trillion human cells in our body. From the moment we are born, we are exiting a sterile environment and entering the world, and we are immediately colonized by a diverse community of commensal microbiota that will grow and develop with you and that you will take with you throughout your life. These microbes live all around the human body, such as the mouth, the vagina, the lungs, and the skin, but the majority of these microbes live in our guts, and they can have great impacts on the health of the host. So some of the ways that the human gut microbiome can affect the host: the gut-brain axis links the brain with our intestinal cells, but because these intestinal cells are also in communication with the gut microbiota, this is a place where bidirectional communication can occur between the brain and the gut microbiome. Additionally, as our gut microbiota grow and feed, they are producing metabolites that can enter the bloodstream and affect distal sites of the body. Previous work in our lab has shown that a large portion of the metabolites in the blood are associated with cross-sectional variation in the host microbiome. In relation to this, metabolites produced by our gut microbiome can also cross the blood-brain barrier and affect mood and cognition. Another metabolite of interest is trimethylamine, which is produced by the microbiota when we eat choline or carnitine, which is a chemical compound that you will typically get from meat or fish or even tofu. But this trimethylamine can travel to the liver through the bloodstream and be oxidized to TMAO, and that is a chemical that is associated with atherosclerosis, or disease of the arteries, and cardiac disease. But metabolites created by our microbiota can also assist with immune system regulation, where imbalances in microbiota, immunity interactions have been implicated with gastrointestinal diseases and immune disorders. And finally, the commensal bacteria residing in our gut can prevent the colonization of pathogenic bacteria via niche saturation, nutrient competition, altering the pH of our environment, or modulating the immune system. So if I were a pathogen trying to invade through the stomach, I would have a lot of trouble because the entire community is already saturating all those niches. There's not any metabolites that I can really take advantage of. So having a diverse gut microbiome can be very helpful. As you may have guessed, we are going to be discussing MacArthur and Wilson's theory of island biogeography. On the right, well, before I start that, island biogeography theory is an ecological theory posited by MacArthur and Wilson that is a classic neutral model of species immigration, emigration, birth, death, and speciation and extinction. On the right is a plot describing the island distance effect, which predicts that as islands get further away from the mainland, the rate of immigration gets lower, and so islands further away will have a lower amount of species at equilibrium than near islands. And on the left is a plot describing the island area effect, which predicts that larger islands have a lower rate of extinction than smaller islands, resulting in these larger islands harboring more species and culminating in a higher amount of diversity on these islands. So what we want to take away from this is the island area effect, and we're going to be applying it to the context of the vertebrate guts. So some research has shown that there is a positive scaling relationship between vertebrate guts, vertebrate body mass, and the Simpson diversity of their guts. So this is a log-log plot, and we see that as vertebrates get heavier, their Simpson diversity tends to be higher. But notably, this data set uses a legacy method of quantifying species called capillary electrophoresis single-strand conformational polymorphism, which is a mouthful, but in essence, it takes 16S amplicons and exposes the single-stranded DNA to freezing temperatures so that they create this unique crystal that is going to travel differentially through a capillary tube, and that's how they quantify the species. So the first step that we did in our analysis was compare this data set to two other vertebrate data sets that use a more modern method of species quantification that we all may be very familiar based on today's class, 16S amplicon sequencing. So on the left, we have the Song and the Grucine data set, and on the right, we have that plot that I just showed you before. And we can see that this signal, this scaling relationship between mass and Simpson's diversity is consistent. The variance explained is hovering around 50%. And this, notably that these plots are both log transformed, and we're reporting this R-squared value from ordinary least squares regressions. But overall, we see that it's consistent. Other methods of species quantification, using other methods of species quantification yields similar results. So the next step was trying to see if this scaling relationship is detectable at the human scale. So here on the left, we have the Aravel data set, and on the right, we have the American gut data set. These are two very large human cohorts, and we have their heights on the x-axis and their Simpson's diversity on the y-axis. What we do see is that the variance explained is much lower than in our vertebrate data sets. However, it's there, it's statistically significant. We're reporting this R-squared using ordinary least squares regression, using the covariates age, sex, bowel movement frequency, body mass and body mass index in order to take away that signal, because we know that those covariates are also associated with Simpson's diversity. And even with all these covariates included, we're still getting that, that signal. And to elaborate on what these other covariates can do to the Simpson's diversity, the health of somebody as they age determines the direction that age affects alpha diversity. So if you are a less than healthy older individual and you're living in an assisted living facility, there is a decline in alpha diversity with age, while healthier older people who are living in the community show a decline in core taxa, but increased alpha diversity with age. As for sex, females tend to have higher alpha diversity compared to their male counterparts, and obesity and BMI are both negatively associated with gut alpha diversity. Finally, bowel movement frequency is negatively associated with gut alpha diversity, with individuals experiencing constipation showing higher gut diversity and individuals experiencing diarrhea showing lower gut diversity. So we have this signal, we found it in humans, and the next step that we wanted to do was reproduce it in silico. And what we do is we created this individual-based model. So this is a schematic of how that model works. We wanted to demonstrate a mechanistic link between intestine size and alpha diversity. So each set of simulations has a bunch of randomly selected parameters: the immigration rate, the reproduction rate, the death rate, and the length of the system. So per time step, based on the immigration rate, there are going to be a certain amount of individuals randomly generated from a log series distributed pool, which is meant to simulate the abundance of bacteria that we see in the gut. So it's not uniform. There is a lot of very common taxa and a tail of increasingly rarer species of taxa. So when we randomly generate individuals, we make it so that they resemble the distribution of taxa in the gut. So these individuals enter the system and they flow through the system 1 unit per time step. And while they're in that system, an individual can be selected for reproduction, or they can be selected for death, or sometimes both. And each individual is just going to keep flowing through the system. And unless they die, they will eventually flow out of the system. And at the end of the simulations, we calculate the Simpson's diversity of the system. So what we did with this individual-based model is we ran 2 sets of simulations, each with 1,000 simulations. But the first one is simulated with lengths that differed by an order of magnitude to resemble the differences in vertebrate sizes. And the other set had lengths that only had a 2-fold difference in the system length in order to resemble the difference in adult human body sizes. And what we can see in our simulated data is that the results are mirroring our empirical data, where the larger scale is yielding a higher amount of variance explained, and the simulations on a smaller scale still have this signal, but the R-squared is much lower. So with that, we can see that in a model, we can replicate what we see in vivo with this positive scaling relationship between body size and Simpson's diversity. So why does this matter? Why would we care? I'm going to go into the clinical implications of island biogeography theory, explaining the variance of Simpson's diversity in the gut. So we do know that lower gut microbiome alpha diversity has been associated with greater susceptibility to enteric infections because your commensal gut microbiota can outcompete pathogenic invaders via niche saturation and nutrient competition. And a relevant enteric pathogen that I think we've all heard of is C. diff. So because we've already gone into it a little bit, I'll keep the background on C. diff infections brief, but C. diff infections, or CDI, are one of the most common form of hospital-acquired colitis, which is a fancy word for inflammation of the colon, and they infect about 500,000 people in the United States annually. Symptoms include diarrhea and colitis, as I mentioned, and disruptions in the gut microbiome, such as taking antibiotics, can make one more susceptible to infection by C. diff, with recurrent illness especially common amongst individuals with low alpha diversity. Another important piece of information before I go into the next set of plots is the impact of diet on alpha diversity. So prior research has shown that diet has a major impact on gut microbiota alpha diversity, which makes sense because this is a huge source of food in the gut, though it's not the only one. But individuals on a plant-based diet tend to have more diverse guts than omnivorous individuals. And this effect has been attributed to a greater diversity of indigestible polysaccharides, such as fibers and complex starches. These can be fermented into organic acids by our commensal gut microbiota, and a subset of these organic acids, such as short-chain fatty acids, have really lovely effects on the gut. One of the more common short-chain fatty acids that we have in our guts is butyrate, acetate, and propionate, and they can help modulate gut pH, prevent the growth of certain pathogenic bacteria, and more. So with this next set of plots, we're using the American gut data set because that data set included C. diff infection history. So on the x-axis, we see if this individual has ever been infected by C. diff. So it's either no, I've never been infected by C. diff, or yes. And on the y-axis in this first row is the Simpson diversity. So consistent with the literature, in the entire group, the mean Simpson's diversity in the no group is higher than those in the yes group. And we chose to separate the groups by their vegetable intake because this is the impact of diet. It also has a great impact on your C. diff, on whether you are going to be susceptible to C. diff. So in the low vegetable intake group, we still see that the mean Simpson's diversity of people in the no group is higher than the yes group. And same thing for the high vegetable intake group. And what we look for in this second group is the differences in their height. So, and the statistical significance of the mean differences in Simpson's diversity is determined by a Welch's t-test. So here in this other row, we have heights. So in the entire group, we see that individuals in the no group have a higher mean height than individuals in the yes group. And this is the same in the high vegetable intake group. However, interestingly enough, in the low vegetable intake group, we see that this difference is not significant, which is pretty interesting. And that leaves a lot of room for interpretation. So next we did a set of regressions, which we show here. So this triangle shows each of the variables I discussed before: diet, Simpson's diversity, and CDI history. And on the right, we have height, Simpson's diversity, and CDI history. So ordinary least squares regression showed that diet is positively associated with Simpson's diversity, and Simpson's diversity is negatively associated with CDI history. Because CDI the responses are binomial, you're either no or you're yes. These regressions are done with binomial regressions, but with diet, we see that diet is negatively associated with CDI history, but height is positively associated with Simpson's diversity, but it is not associated with CDI history in our binomial regressions. And the next thing that we wanted to do is conduct a causal mediation analysis. This is a statistical modeling approach that can, excuse me, help determine if there is a third variable that explains the association between an exposure and an outcome. So in this case, diet is the exposure and CDI history is the outcome. So in the case of diet, we have these values here, the average causal mediated effect. So this is the mediated effect of diet on CDI history, the amount that Simpson's diversity is mediating the effect of diet. So we do see that this mediated effect is present, it's negative, excuse me, and it's statistically significant. The direct effect is also present and statistically significant. And overall, the total effect is negative. But we can see that diet alone affects CDI history, and diet via Simpson's diversity, it's affecting CDI history. However, in the case of height, we only see that this mediated effect is statistically significant, while the direct effect and the total effect are not. And so a possible interpretation is maybe height's effect on CDI history is completely mediated by Simpson's diversity. And that might be why we're not seeing that effect in that low vegetable frequency group, because the effect of diet is so much stronger. So what we want to take away from this is, if you're a shorter individual, you can rescue your phenotype or that negative effect on Simpson's diversity via changes to your diet, which yields much stronger results. So, conclusions. We see that height is positively associated with Simpson's diversity at the human scale. We see that height's effect on C. diff infection history is entirely mediated by Simpson's diversity, and individuals of shorter stature can mitigate the negative effects of height on alpha diversity via changes to their diet. Some references for the images that I used. And finally, acknowledgements. So I'd like to thank the Gibbons Lab for providing support and feedback for my research experience. I'd like to thank Ken Losey for co-creating the individual-based model that we use to create those in silico simulations. And I'd like to thank Florent Mazel for providing the song dataset, one of the vertebrate datasets that we used in those vertebrate plots, as well as the Washington Research Foundation and the NIH for funding my research. And that's all I have for you today. So I'm open for questions now.
Yeah. Great talk. Yeah, really appreciate you sharing your work with us. That was really very interesting. We've got a couple questions in the Q&A that people were wondering. So I'll start with the first one here. Did you see any diet effects within the vertebrate animals that you tested?
Yes. So in our animal data set, we can see that whether they're carnivorous, herbivorous, or omnivorous. And in each respective data set, we see that there's a significant association with diet. However, it's not consistent. So if you're an herbivore, the effect of herbivory is different. It's in the opposite direction in one data set versus the other. So that's interesting. I'm still trying to kind of look into that more and figure out why that might be. But yes, we were interested in the impact of diet in our vertebrates as well.
Very cool. And very cool stuff. All right, another question that's just popped up. What kind of diets do you think would mitigate the effects of height?
Eating more vegetables, I would say, or eating more plant-based foods, because as we said before, all the fibers and the complex polysaccharides are very nice fuel for the commensals to make some pleasant chemicals in the gut.
Super interesting. I think that's what your data backed up as well, is that more vegetables seem to be a good thing. Another question, do we know if height and gut length are actually correlated?
Yes. So the literature can be mixed at times just due to different methods of measuring the different parts of the gut. But as a whole, yes, height and gut length are positively associated.
Very cool. Okay. So another question that's just come to the Q&A. Will eating vegetables be applicable to all races in affecting their heights?
So in our data sets, I did not include race as a covariate, but I would argue that a lot of science is showing that race is more of a social construct. And I would say that I think everybody, no matter what their race, would benefit from eating more vegetables.
Cool. And actually something that I was just wondering, I know you said that, eating vegetables might rescue the phenotype for individuals that might be a little bit shorter. Can you sort of conjecture on what these results might mean for people who are on the other end of the spectrum, who are already taller? Do they not have to worry about eating vegetables?
Who are taller, I would say they should still eat their vegetables, just because the effect size of height is pretty small. So as compared to diet, so I think there would be still benefits to those taller people, to the alpha diversity of their guts, even though they are winning in that regard and getting a little boost in their alpha diversity.
Totally. All right. Well, it looks like that's all the questions that we have.
I think I see one more.
Oh, yes, sorry. A couple more have just popped up here. Yeah. Was there any evaluation of the DNA impact on the microbiome?
I can.
Maybe you can.
Can you rephrase your question? Maybe I think I don't understand.
Yeah, perhaps you can rephrase your question, and we'll see if we can answer that as well. But while you do, do we think the effects of diet would be personalized as they are for SCFA production in terms of non-responders or even the loss of diversity?
Yes. Yeah. So previous research by our lab has shown that there's a subset of people who, even though they're eating more fiber, they're not responding with more SCFAs. So there's definitely some room for more interpretation in that regard. But yeah, that would be very personalized as well, based on your gut microbiome.
Okay, so we have a clarification on the previous question. For humans, DNA SNPs have been shown to impact the microbiome. Yeah, I think that's been determined. I don't know, Kat, if you have anything to add within the context of your work.
Yes, so individuals' genotypes were not used in my analysis, but that would be a really interesting way to further look into this. Like maybe that's the key for why we're seeing that height association disappearing in that low vegetable group for CDI history. Yeah, thank you, Ken.
Well, thank you so much for sharing your work with us here today, Katherine. If there's any more questions, feel free to reach out on Slack, and I'm sure we'd be happy to answer those questions as well. But yeah, thank you so much for taking the time to share your work, and thank you to all of our participants for listening. And now we'll have Christian Diener will be giving us some closing notes for today's part of the course. Here, and change your. Oh, okay, yeah, it should be working.
Same system.
Yeah, I think it's fine.
Sorry about that. I think we had some audio problems. I hope everybody can hear me well now. Yeah, I'm good. Yeah, no, I'll start from the beginning. Apologies for that. So yeah, as I was saying, that about wraps it up for day one. We're a little bit ahead of schedule. So even with that little bit of audio problems, we're still like pretty early in our schedule. So what I was doing before is that I was starting to thank our sponsors, Applied Microbiology International, Illumina, Metabolon, and the University of Washington Embrace Center here in Seattle. Without their contributions, that event wouldn't have been possible. So we are very grateful for that. We also got some notes that I think the live stream on YouTube crashed at some point. So in case you were joining that, just know that all the videos like of the presentations and the symposium will all be posted pretty quickly, and we will let you know on Slack. I think the course video from today will be posted between today and tomorrow. So you will be able to get back to that and also look at it on YouTube. And they will also be posted on the event website, and we will let you know on Slack. So for today, I would like to thank specifically Nick Quinn Bowman and today's presenter, Katherine Ramos Sarmiento. We all learned a bunch from them, and it was like super cool to see the course like go pretty much without a hitch, except for a small little snack on YouTube. So just to recap what we learned here today, we introduced you to like a computational tools for amplicon sequencing data analysis, and also had to go into the nitty-gritty of analyzing some data from healthy individuals with a recurrent CDI infection. If some of the course seemed maybe a little bit too fast, or you got stuck on some part of the notebook, just know that you can feel free to come back to that at any point. You could watch the videos, you can go through the notebook again. The TAs will be available on Slack for a long time after the event has ended. So even if, for instance, you didn't find much time today and you want to get back to it in a week, just feel free to do so, and we'll be able to help you there as well. And that also applies, for instance, if you were in a different time zone and this course happened to be in the middle of the night for you. So yeah, feel free to take your well-deserved time to sleep and then get back to the course whenever you feel like it. So if you want to share your experience on social media, the hashtag for the event is ISB Micro23. If you want to tag ISB, the handle of the ISB is ISBSCI. So we'll be happy to hear what you thought about the events, or if you have any other questions, also feel free to use those channels. We look forward to hosting you here again tomorrow. We'll be having the second part of the course taught by Alex Carr, And he will actually show us how we can use that data that we generated today to actually start building mechanistic models of those microbial samples, and then ask questions, for instance, like how susceptible are those samples for a pathogen to come in and actually engraft stably, or are they actually protective for that? So we'll be seeing that tomorrow, so feel free to tune in again. We will kick things off at 9 a.m. Pacific time. And again, we will also give you a heads up on Slack before we actually start the course. We hope to see you there. So it will be, I think, a really cool application of the data you generated today, and actually, like, you know, playing around with them a little bit more and getting some biological insights as well. If you want to learn more about the ISB, you can do so at our website, which is isbscience.org. And yeah, thanks so much, everybody, for joining us. It was great engaging with you also on Slack and social media, and I hope to see many of you tomorrow. And thanks again, and bye.